Okay, so I am Alex Putman with University of California Riverside. I'd just like to welcome everyone here to this workshop on INSV. And so our goal of this workshop is to, give a overview of the important points of this virus with respect to lettuce in preparation for the upcoming winter produce season in the desert. And so we have a pretty good a panel here in terms of who is speaking. And just before we get started, I'd like to really thank Stephanie Slinski, who really came up with the idea and did a lot of the work to organize this workshop. All right, with that, let's get right into our first speaker. Is of course Stephen Koike. He is a plant pathologist for and director of TriCal Diagnostics in Hollister, California. He and his team operate a plant disease diagnostic lab, conduct research on soil-borne and foliar pathogens, and provide plant pathology expertise to clientele. Previously, Steve was the California Central Coast Extension plant pathologist with the University of California Cooperative Extension. He has published extensively, including his 450-page book, Vegetable Diseases, A Color Handbook. Koike has received numerous awards, including the National Excellence in Extension Plant Pathology Award from the American Phytopathological Society, and the Outstanding Contribution to Agriculture Award from the California Association of Pest Control Advisors. All right, take it away, Steve.
Thank you, Alex, and good morning, everybody. All right, well, first, I also want to thank the YCEDA group and Stephanie, Alex, and the committee for inviting me to participate in the workshop today. So it's my pleasure. I thank you for that. And I know a lot of you in the audience probably have a lot of experience, not only with lettuce, but with INSV, which is Impatiens Necrotic Spot Virus. So for those of you who do have that experience, some of this will be a review for you, but hopefully together we'll reinforce some of the dynamics with this important problem. And we'll all learn some new things. And we have, like Alex said, a really excellent group of speakers this morning. So I'm pleased to be a part of that. So let's get right into this. My assignment is to briefly talk about the history of this problem on coastal California lettuce, and then I'll focus most of my time on some of the diagnostics of this. So again, for many plant diseases, some diagnostic tasks are very simple and straightforward. INSV can be that way in some situations, but there are a lot of complicating factors that make it a challenge. And I do diagnostics 100% of my time. That's my career and time. And we are sometimes confused by what we see. So I like to share some of that information with you about overcoming these challenges, or at least knowing what factors are involved with diagnostics. Okay, so we'll get right into it with the history first. So a lot of you probably know this already, but before 2006, we did have a Thrips vectored virus on lettuce in coastal California, but it was not INSV. It's actually the other important one, tomato spotted wilt virus. I think Alex or other speakers may go into this, but in California, there are two main Thrips vectored viruses that affect us. One is tomato spotted wilt virus, the other is INSV. They're closely related, but they're different. But for lettuce in California, before 2006, we definitely had tomato spotted wilt around, but very unimportant. If I found one or two plants in a year, I'd get really excited and take a dozen photos because I'd know that'd be the only time I'd see it on lettuce. So it's really a non-factor, but it was around. Interestingly, 2006 is that pivotal year because that was the first time we had INSV on lettuce in the Salinas Valley. And then the next couple of years, it was not just a one-time thing. 2007 and 2008, we saw an increase in that, very incremental. I gave a range there. Most of the fields we looked at those first few years are way below 1%. We had one or two blowouts, which made it interesting and turned it into a research project for us, where we had maybe half the field with it. But those are outliers. In those first years, we had very low incidence, but it was moving around. The 2008 to 2017 period is very important to note because we had a slow but steady increase of INSV on coastal lettuce. And I like to highlight that period for you, 2008, 2017. That's when we feel there's clear evidence INSV became established in the Salinas Valley. So it was no longer just a passing thing, interesting disease development, but we saw during that period of time, every year we would have it in some areas the same ranch year after year. New ranches, new areas would come up the next year and subsequent years. So that period of time is very important in understanding the dynamics in California coastal lettuce, because it became established during that period. And of course, most of us are on this talk and Zoom today because of the 2018 to 2021 period, where there's now a significant increase in INSV, and not only in incidence and where it's showing up, but in impact. We're having a very important crop loss situation now with INSV in the last 3 years or so. So with that brief history, I hope you see that this is not a brand new thing. Even though 2018, 2019, and 2020 had really important outbreaks and crop loss with INSV, we've had this thing here now since 2006. So it's not a brand new development, but you could see how it's increased in importance over that period of time. And then part of the history now includes these complications. And I'll go into this quite a bit in the last half of my talk. But we have Pythium wilt and other diseases that affect lettuce. And we're having co-infections. We're having more than one pathogen on any one plant in any one ranch or block. And so that complicates the diagnostics of it. And I think all of us as plant pathologists and researchers would agree that increases the risk of loss and the crop loss and the dollars lost from not only INSV, but these complex and multiple infections. And then finally at the bottom there, not my assignment to talk too much about this, but just to be complete, as you know, we are now finding INSV in limited amounts, I think, in some of the desert counties in California, and of course in parts of Arizona, primarily Yuma, but other places too in Arizona. So we're sharing the wealth with everybody in Arizona. I wanted to put together kind of a graphic to illustrate the trends and patterns I just talked to you about. So these lines are timelines, and the thickness of the line indicates the importance or the relative importance of the problem. So let's look first at the two blue lines at the top. Tomato spotted wilt virus, again, very close relative of INSV, both spread by thrips, but it's a different virus. And I joined UC Extension in Salinas in 1989, so that's kind of the endpoint on one side, and then to today, current situation on the other. So you could see in the blue lines, for horticultural crops and other vegetable crops, we've had tomato spotted wilt forever. From before 1989 through the present, horticultural crops, greenhouse crops, and other crops in the field, tomato, pepper, other things, have always had and almost always will have tomato spotted wilt. And the line is fairly thick. I would call that a low to moderate level of importance for these other crops, horticulture and other crops. The blue line below that is lettuce. And I mentioned before, we do have tomato spotted wilt on lettuce. It's been around a long time, but I hope you see in the visual, it's a very thin blue line. It's not important. We have a hard time finding it on lettuce. When we do find it, it's a few plants. So for tomato spotted wilt on lettuce, not a factor, but it's been around for a long time. Let's drop down to the green lines down below. This is INSV, and it's very much the same as the tomato spotted wilt situation for horticultural, greenhouse, and other field crops, other vegetable crops. INSV has been around in the Salinas Valley and the coastal part of California for a long time. It was here before I came, and looks like it'll continue to be here for a long time after. So you can see that low to moderate level designation for that top green line for horticultural crops, greenhouse crops, and vegetable crops in the field. INSV has been around and for a long time. The puzzling thing is for lettuce. Lettuce did not have a record of INSV until 2006. To our knowledge and to all research information and publications, we have no record of INSV on lettuce until 2006. And why that is still escapes all of us who are doing research on this virus. The virus is around, it's in the Salinas Valley, been there from before 1989. Why did it jump to lettuce early on? We don't know the answer to that. And real quickly as an aside, The strain of INSV that affects greenhouse crops, chrysanthemums, lisianthus, other crops, it's the same strain that goes to lettuce. So that is not the answer in terms of a strain difference. In addition, you could see the way that line increases over time. It's just not a straight line where it's the same every year. As we mentioned earlier, there is a significant increase in incidence and importance from 2006 to the present. So this graphic shows how lettuce is an outlier. It looks very different in terms of the importance of INSV on this crop compared to other INSV hosts and compared to tomato spotted wilt virus. Okay, so let's move on now to diagnostic considerations and how you diagnose and identify INSV in the midst of all these challenges. So first, we want to say that it's been misdiagnosed from the very beginning. From 2006, there were cases of being misdiagnosed because it looks like other things. So that is the kind of legacy of this disease on lettuce. It's easily misunderstood or misdiagnosed as something else. And that misdiagnosis is still happening today. In our lab, we receive hundreds of lettuce samples, hundreds, and many of them are INSV. We have some very, very sharp PCAs and growers in our region, and they understand this disease pretty well, I think, but there are still cases where it's missed or misdiagnosed for something else. So even trained, experienced professionals can make mistakes, including myself. And then the other consideration, which I'll go into with you this morning, there are these complicating factors that make diagnostics, more of a challenge. We have other viruses that look similar. We have co-infections with fungal pathogens that muddy the diagnostic water. I'll go over those with you. And then abiotic factors, primarily chemicals, herbicides, can cause damage that looks very similar to INSV. Okay. So with that setting the stage, I'd like to show you some examples with photographs and case studies of INSV diagnostics, and let's look at these together. So first of all, here's a photograph of spray burn. And I know you in the audience, you PCAs, you've never burned lettuce before. So you probably don't know, I don't recognize this. I'm joking with you, of course, but spray burn on lettuce is a thing. In this case, it was caused by an unnamed PCA. We will not reveal the name to protect the guilty. Oh, I'm sorry, my mistake. This is not spray burn. This is INSV. And actually, this is a very historic photo. I want to show you, this is the very first, to our knowledge, our very first picture of the very first case of INSV in 2006. So because we're all friends here, I just want to show you this very historic photo. Sometime Stephanie will print this and frame it in your office. The first California case of INSV on lettuce. And interestingly, this was misdiagnosed by an unnamed extension plant pathologist in the region. We don't need to go into the name. We don't. It's not important to know who did this. Although I kind of wonder what the University of California is doing, hiring people who can't diagnose diseases properly in the field. But this is INSV for the first time on lettuce in California, and it was misdiagnosed for a period of time as chemical damage. Okay, let's go to INSV. Picture of INSV on lettuce. You can see the browning, the burning, looks like spray burn. Oh, I'm sorry, this is another mistake. I'm sorry about that. This is lettuce dieback disease. This is another virus problem. I think many of you probably are familiar with this. It's caused by the virus pathogen tomato bushy stunt virus. A very odd virus. It's soil-borne. It's in soil-borne. It's a soil-borne virus. It affects primarily romaine and leaf lettuce types. It does not affect head lettuce. And again, as I mentioned, it's in the soil. If you know anything about virologists, they like to give these pathogen names that have 3 or 4 or 5 names or words in them. So this originally, when we started working with it, was called tomato bushy stunt virus. It was renamed later as lettuce necrotic stunt virus. Bill Winterman, another virologist, found that this was related to something else that's renamed Moroccan pepper virus. I don't know, Bill, if you're on here, but we're digging at virologists a little bit here. And if Bill ever gets it together, we're going to publish our paper on that. Currently, we're calling it lettuce dieback associated virus, but I think most growers know it as tomato bushy stunt. But I hope you'll see that this looks like INSV, and so there's a chance it could be misdiagnosed. Okay, how about this? Well, finally INSV, right? You can see the necrotic lesions, burning. Okay, I'm sorry, another mistake here. This is actually chemical damage. This is bona fide confirmed herbicide damage on lettuce. It looks very similar to INSV. If you have experience with INSV, you know that the symptoms overlap quite a bit. There are clues to help you in the field. Again, you know this already. If the weeds in the field or the weeds around the field have similar symptoms, you have to start thinking about other factors. Now, it could also be virus, but you need to check into the possibility of it being chemical, drift, herbicide, something like that, if you have the same symptoms on multiple species, including the crop. And then, of course, field patterns are important to analyze. As you know, sometimes the drift will be worse along an edge, which is the source of the drift or the spray. It'll subside and become more dilute as you move into the field. There are these patterns you want to look for, right? So that will help you separate chemical damage from INSV. How about this? You know, if I was in the same room with Alex, I would quiz him and make him cough up an answer on this. But what do you think about these two heads of lettuce? If I were you, I'd start be thinking about physiological problems. There are physiological head disorders on lettuce that look similar to this. Because of time, we'll move into this quickly. We do have INSV here. The bottom left photo is INSV on head lettuce. But the top right one is cucumber mosaic virus on lettuce. Now, I don't know about you. I spend 100% of my work time as a plant pathologist. I'm trained in these things. I would not venture a field diagnosis on whether the top one is INSV or not based on the symptom. We've seen dozens of head lettuce varieties that look similar to the top right photo, and that's INSV. This one in particular, one pictured here, is CMV, so it's a different virus. So again, diagnostic confusion because there's overlap of symptoms with different viruses. How about these two, Alex? Again, I would pick on him if he were in the same room here. Again, because of time, let's move into this. So we do have INSV here. Top left photo, INSV. Again, to me, that could look like spray burn or other things. Maybe not tomato bushy stem as much, but possibility. The bottom right one is tomato spotted wilt. And again, tomato spotted wilt on lettuce is a thing. Not very important, rare in most situations, hard to find, but it causes identical symptoms. These two viruses cause identical symptoms on lettuce. Okay, and then I'm through messing with you now. So here are four photographs of INSV on lettuce, just to give you a range of symptoms. Again, you get what we call necrosis, which is brown tissue. Necrotic dieback of the leaf tissues, there's spotting, there's deformity. We get symptoms on both old and young leaves. That's an important feature to match. Sometimes, like on the bottom left, there's a hint of ring spotting or spotting. We don't see that on all cases, but these four photographs, I hope, show that there's a range of INSV symptoms. And if you really need to know if it's important to have a precise identification, you have to test. Okay, you can't guess. So in summary, in terms of the diagnostic features of INSV on lettuce, I hope the photographs show that the symptoms can vary. And it's like any virus infection, lettuce is not unique in this case. The symptoms will vary based on various factors. And that could include the age of the plant when it was infected, the type of lettuce, the lettuce cultivar, the environmental conditions. Depending on the weather and the environmental conditions of the crop, that influences the expression of virus symptoms, so that could be different. And also the virus level in the plant. If you have a plant that's been recently infected and it's not gone systemic and you don't have a lot of virus in the plant yet, it'll look different than a plant that was infected young and the virus now has inundated all the tissues and is everywhere. I have virus strain in parentheses there. As we all know from COVID, Delta strain, in viruses, the strain can be very important in terms of symptoms and the disease. However, for INSV at this point, we have no evidence of different strains. So until other research or other investigations show that there are differences in the strains, Our work, at least up to now, shows there's no differences. The inosmine lettuce is all the same. It's also the same as the one that goes to chrysanthemum and tomato and pepper. So in terms of the strain, it appears not to be a factor in the variation of symptoms. Then the other things to remember, INSV symptoms overlap with those of other viruses. So you have to keep that in mind. We have other viruses still important out there, and you can have those causing similar symptoms. And then, of course, with chemicals, especially herbicides, you can have, or fertilizer overdose, you can have abiotic factors that cause similar symptoms, and those need to be considered when you're looking at your field diagnostics. Final comment about the lettuce. This does not apply to lettuce. Some INSV hosts do not make symptoms, and I think Daniel or some other people might mention this later, but some of the weeds in particular, they are apparently very important virus reservoirs of INSV, but they are sometimes symptomless. They do not show symptoms. Lettuce is not one of those hosts. As far as we know, when you have infected lettuce and the disease is developing, you get symptoms, but that's not true for all hosts. And speaking of other hosts, I won't spend a lot of time on this, anytime actually, but just keep in mind there are other commercial crops out there that are hosts of INSV. We find a number of these in Salinas Valley. You in the desert should be looking for these as well. And as you go about developing your expertise in identifying these or recognizing the possibility of INSV, you need to be familiar with some of the general symptoms on those other crops as well. Bottom line is, if you need to know if it's important, you must test. You must send it to a lab like ours or do other tests yourself to investigate whether it's INSV or something else. Okay, in the time I have left, I want to address the big topic of co-infections or multiple diseases on any one lettuce plant. And it's pretty amazing to me. I don't know, maybe you're not surprised, but I'm amazed at some of the crops or some of the plant samples that are submitted to our lab. There are cases where they have 3 different pathogens on one lettuce plant, 3 different pathogens on one plant. And we can separate those by testing. In the field, I think it's more challenging, and there are places where you can make mistakes if you're not looking for multiple infections. And then the multiple infections can also complicate the diagnostic process just in general. So I'd like to briefly talk about that and demonstrate that to end my time with you. So this works for me. I don't know if it works for you, but what I'm doing and what helps me in the lab here at TriCal Diagnostics is we definitely visualize and conceptualize and consider lettuce plants now in 3 parts, 3 distinct sections. And we analyze each of the 3 separately. We don't assume anything. We consider lettuce as having 3 parts that need to be looked at independently. So part 1 is the top foliar part, above ground part, basically. Part 2 is the middle part, which would be the crown and the main core or vascular tissue inside the crown. And the bottom part, the third part, are the roots. And at least in terms of my work, it helps me to consider each part of the lettuce separately. And so we look at each part individually, independently, and we do not make assumptions about that. So let me show you how we kind of approach our diagnostic strategy with that, with some photographs here in cases. So again, this is all with co-infections in place. So first of all, we're going to look at just single infections. So if you have INSV only INSV in lettuce, you're only going to have symptoms in the top part. The foliar above ground part is basically where you have the INSV symptoms. The crown and the vascular and the roots will not show symptoms directly from INSV. And, you know, I'm going quickly here because of time. There are different nuances here. You know, if you have an INSV infected plant that's been out there for a month and a half, it'll start to decline. You may have other things start to happen below ground. So of course we can discuss that in questions or other times, but in general, INSV only is only a top symptom deal. It does not affect crown, vascular, or root. And I think a lot of you are very familiar with the crown part. If you have crown only, crown infections only, which is primarily Sclerotinia and botrytis, of course, the crown is going to have the main part of the symptom, and it's going to be severely affected. But with Sclerotinia and botrytis infections, you do not have root symptoms. It is not a root pathogen. And then the top will be affected because you've taken out the crown. So when you have a diseased crown, the top part of the plant, of course, will show symptoms, but not virus symptoms. It'll be collapsed symptoms, pretty straightforward, with only sclerotinia or only botrytis. Same picture with the wilts. If you have verticillium or fusarium wilt, it affects the main core, central core of the lettuce plant, but the roots will be healthy. It is not, these things are not root pathogens. Verticillium and fusarium directly will not cause root symptoms. The tops will be affected, but because of the collapse of the crown and vascular tissue. So you'll have the collapse of the top part. And finally, again, these are single case infections. If you have root issues, and in coastal California, we're very concerned with this Pythium wilt disease. I think I may have talked about this with you desert people in previous meetings. But Pythium wilt and also Thalaviopsis, another root rot organism on the coast here. These are very important now, becoming very important on California coastal lettuce. But if you have the Pythium wilt disease, it only affects directly the root. The crown will be healthy, and then of course the top will start to decline because you've taken out the roots. Okay, so why have we taken the time and effort to dissect lettuce in these three parts? It's because, in my opinion, at least in terms of how I approach things, for me to be more thorough in my diagnostic chore and to miss fewer things, I need to consider lettuce in the three parts: the top foliar, above ground, the middle crown, the bottom root. And in my opinion, when I divide lettuce into these three parts, there's fewer chances I'll miss something. If you don't do this, I believe there's a more chance you can miss something. So here's my first example of that. We get plenty of samples that come into the lab here that have both INSV and the sclerotinia or botrytis problem. Now, I can't really show you a good photograph of that. I went through my hundreds of photos and didn't really have a good example of the top co-infection, but I would like for you to visualize the merging of the two top photos. When you have INSV symptoms in the above ground portion and the plant starts to collapse and melt before your very eyes, it's not that easy to pick out virus symptoms in a collapsing plant. Again, if you're good at that, I applaud you, commend you. For me, when I have a collapsed, oozing lettuce head, it's sometimes hard to see the virus symptom amongst that decay and collapse. And so again, my point is there's an opportunity there or a possibility of missing the virus because of the crown infection and the collapsing head. So if you have a co-infection with a crown pathogen and a virus, you can miss something there. We'll go through these quickly. Same principle for verticillium fusarium. We have dozens of examples of a vascular wilt problem. It's causing the top to collapse, but it has virus as well. If you merge those two top photos together, it's not that simple to pick up the INSV. It can be missed. The roots, again, will be healthy. And then we see a lot of this. This is the probably #1 combination we're seeing right now in the coastal lettuce, Salinas Valley. We have a lot of this Pythium wilt occurring. And when you have Pythium wilt, the tops will collapse. Merge those two photos together, and it's hard sometimes to pick out the virus if you have a collapsing head. So this is with the co-infection of a root problem with the virus. And I think I mentioned at the very beginning of the meeting, we definitely have cases where you have three problems on one single plant. Again, to me as a pathologist, that's kind of amazing, quite alarming actually, but you have the roots gone because of Pythium, you have a crown or vascular problem in the center there, and then it's a mess on top. You have collapsing plants with virus symptoms somewhere in there. When you have that combination, it's sometimes hard to know if there was virus in there at all. So my suggestion to you again is to strategically divide your lettuce into three parts: the top part, middle crown vascular, and the root, and you'll have fewer chances of missing something. At least that's how we approach it, because we don't want to miss things for our clients. So as I wrap up here, some summary statements about symptoms on lettuce. So if you have INSV only, this is only INSV. This is going back to this photograph here. If you have a series of photographs, if you have INSV only, You will not have wilting. You will not have wilting generally. Again, the footnote here, advanced cases, plants are declining, it's dying. This changes the situation, but in general, you will not have wilting with virus only. You will have no root rot. The roots will look great. There'll be no discoloration in the roots if you have INSV only. The crown will be solid. You can kick that plant. It's not going to fly 6 beds over because the crown is decayed from sclerotinia. No crown rot if it's INSV only. No vascular discoloration. Dig up that plant with the virus only, slice down the crown and vascular tissue, the core. You will not see any discoloration from virus only. And then we've repeated this a couple times. Keep in mind that these symptoms on lettuce definitely resemble chemical burn. That unnamed extension agent who misdiagnosed that in 2006 made that mistake. It looked like spray burn, and that's what he called it. And then keep in mind, hopefully I illustrated this sufficiently for you, the INSV and solar and pathogen thing is a big deal in the Salinas Valley. Maybe you're not seeing that in Yuma or Desert California counties. Hopefully you won't. But for us, we have these complexes, these co-infections, and it's a confusing combination of symptoms. So as I wrap up here, we like to illustrate and emphasize that these co-infections influence how people sample. So again, we get hundreds of samples of hundreds of samples of lettuce in our lab here at TriCal Diagnostics. And our customer base, I think, are very experienced. For the most part, they send in very good samples. But there are cases where we'll get partial samples of a lettuce, as illustrated on the 3 photos on the left. I think what happens is some growers, maybe some PCAs with less experience, they'll go by and see lettuce, and they'll say, that looks like a virus. I need to know if that's INSV. I'm going to collect that for Steve, and they'll send that in. What they do is they cut the lettuce off at the ground level, and they'll send in the head. They do not send in the crown. They do not send in the root. And so those grayed out areas represent sampling error. We're not seeing these parts of the plant, and therefore we can't see if there are possible co-infections. And I think in these days, information is very important for your work. The more information, the better. If you're missing information, that puts you at a disadvantage in terms of doing your job to the utmost perfection. And really, to be honest, from a business point of view, if you're missing things, your competitor might pick that up and have an advantage over you. That's how I look at it. So we want a sample on the right with those 3 photographs, meaning we have the top, middle core, and root sample. You've got to collect that carefully, dig it up. You can't yank it out of the ground, but send in the complete plant to cover these 3 bases, which I think is essential now for not only lettuce, but all plant diagnostics really, but really important for lettuce these days. Okay, and with that, I'm done, Alex, and I think I'm passing it back to moderator or someone else with that.
All right. Thank you very much, Steve, for the excellent presentation. So just a reminder to everyone, the poll is open now. And so, take a look at that as we answer questions. And as we get into the end of the Q&A session for Steve, we'll give a notice, and then we'll close the poll. And also a reminder for those online, please submit your questions to the Q&A function. And we have a couple questions already, and we're going to alternate between online and in person. And the first question is from Ryan Kelly, and that is, in what area of the valley was the first case discovered?
Oh, great question. Of course, we will not name a ranch, right? To not incriminate anybody. I'm trying to visually see that. That is, it was south of Chular. Yes, the south of Chular region on the east side, so east of the freeway. And I can't remember how bad it was. I think it was like a pocket, kind of an edge effect where maybe the thrips were congregating. And the grower, I may have told this story before, the grower brought it in, and he was frankly quite alarmed because he was being blamed for burning the lettuce. And he said, Steve, there's no way. I mean, didn't spray it. I know I didn't burn it. Now, last year, I did burn that lettuce, but not this year. So, and that's where, again, that unnamed farm advisor caught a spray burn until more work was done. Good question.
Okay, thank you. Now, Stephanie, do we have a question in person?
Do we have a question? No, doesn't look like we have a question here.
Okay, we have another question online from Robert Chenoweth. Are you able to detect INSV in symptomless roots?
Huh. That's a great question. I have not tried that. So I would assume the tests are serological tests, so they should pick up virus if it's present. So I would assume, and that underlying assumption, that the answer would be yes, that we should be able to do that. Be interesting to try that. I have not done that.
Okay. We don't have any other questions right now, but I have a question. So Steve, you were talking about how, TSWV, for instance, and, it was just kind of constant at a certain level for a long time on the coast. And to clarify, was that on the coast? And then also during that time, was TSWV a big problem in the Central Valley on tomato?
Okay. So this is all coastal information, Alex, if that's the first part of your question. Mm-hmm. So yeah, so I don't know the status of these things in the other valleys. The other thing I could add is on lettuce, there's that smaller industry in Huron, and Tom Torini, the farm advisor there, worked with us on this project. We never found INSV on lettuce in the Central Valley and the Huron lettuce industry. We did find tomato spotted wilt. I don't believe, we'd have to check with Tom. I don't believe it was a really severe issue for lettuce in that area, but it was present, but no INSV.
Okay, thank you. Okay, we have a couple more questions came in. So Junior asks, you might have covered this, and I don't think you did, but how long can it take for the lettuce plant to show symptoms of INSV? I think Daniel might touch on this later as well.
Yeah, I think other people can do a better job. I mean, there's always a latent or a lag time, as you already know. So once it's infected, it takes, 5 to 7 to 12 days maybe before you have expression, but that's just a ballpark figure. Perhaps other people have better information, but yeah, it definitely takes a few days up to a week before you'd see anything for sure.
Yeah, good question. Okay, next question from Martin is, can INSV be diagnosed using molecular diagnostic tools?
It certainly can be. Yes, the answer is yes. The serological tests are quick. There's these immunostrips that they're not super sensitive, but they work. And then there's ELISA tests that can be done as well. Again, maybe Daniel and others have more information, but certainly there are molecular methods. But I think serological tools are easy and available, and so they tend to be the first ones used, I believe.
Yeah, so to follow up, are there cases where, you or other diagnosticians need to be using those, or think is the serological, the strips or the ELISA are basically, sufficient in most cases?
Yeah, good question. We always want to consider that, as good scientists and diagnosticians, you don't want to make assumptions that that's the only way to do it. I think until we have other indications, it seems that covers the diagnostics quite thoroughly. So I don't think we're missing anything, but that's always a good consideration to have just in case.
Okay. I think this question from Andor was similar to a previous question. And next question from Troy, why is INSV not showing up as heavy as last year? I think others may touch on this as well.
Yeah, I think so. I mean, we think the weather, it's all predicated on thrips, right? So if the weather's cool, the thrips population doesn't blow up as soon or as extensively. So that's probably part of the answer. But yes, others will have better information, more specifics.
Okay. Yeah, that's a good question too. And I think next question from Daniel, I think is a big question. What do you think has changed from 2006 to have INSV in lettuce?
Oh, wow.
It's the $1 million question.
Yeah, excellent question. I hope the questioner has an answer for me, because we'd love to know. And you and I, questioner, we can publish that. Okay. We don't know. I mean, I made a comment about establishment. That's just kind of a summary based on our research. But we feel the virus became established in that period of time, 2008. to now. And so that increases where the virus is in the valley. When a pathogen is established, there's reservoirs. It's everywhere and becomes kind of ubiquitous. And so I think that's part of the answer, but we really don't know exactly why. My big question is why not before 2006? I'll probably never have an answer for that, but the virus is present. We have. I did this myself. I went to greenhouse crops in the Salinas Valley inside the greenhouse, got INSV from various cut flower crops. It's the same strain as the one that goes to lettuce. Why didn't that show up in lettuce before 2006? So, excellent question. Thank you.
Yeah, very good question. Okay, so we'll do one last question before we talk about the poll. So, do any abiotic conditions increase virulence of virus infections, such as hot, wet, or windy conditions?
I would say no. Again, I would welcome other speakers to touch on this if they have time or the inclination. I would say that doesn't really work for viruses. I think it's just a dynamic of the Thrip vector and a susceptible host. I think that's what's the key. I don't think the condition of the host or abiotic factors or stress affect that, and which is very different than other things, right? Like Pythium on lettuce. The wetter the soil, the more predisposed there is for disease, because Pythium likes the water. I don't think that works that way on viruses, but I certainly can stand to be corrected if there's an expert that knows more. Good question.
Yeah, good question. Okay, so I think there's a couple questions here that I'm going to touch on, and maybe we can have this Steve maybe answer this question, and by typing it in a minute. So we're going to wrap up the poll in about 10 seconds, and then give Steve a chance to go over that. Okay, there we go. Steve, can you take a minute to go over and just highlight any questions that people had difficulty with?
Okay. I'll be brief because of time. But yeah, the C, of course, was the correct one here. It's been around a long time, but only recently has been affecting lettuce. Number 2, yeah, I mean, that's the mistake that that unnamed farm advisor made. Definitely looks like spray burn. I think very few people would have a problem with that one. Number 3, yes, that's a problem for people studying the virus, trying to find it. A lot of the hosts, especially weeds, do not show symptoms. So that's kind of a bothersome thing. You have to test things that don't look like they have a disease, and how much of that can you do? Number 4, the true statement is that yes, we definitely have virus-infected plants with fungal pathogens, and that's a big challenge in terms of the diagnostic challenge. And then finally, number 5, yes, the co-infections mean that all plants, all parts of the plant should be examined. And if you're submitting a sample to our lab or some other lab, you want to do that anyway for all cases. Submit root, crown, and top. That's always the best way to go.
Okay, excellent. So yeah, thanks again, Steve. Great job, everyone, on those questions. And all right, we'll move on. And so I am the next speaker here. And so just a bit about myself. I'm a cooperative extension specialist in plant pathology at the University of California Riverside. My program focuses on vegetable and strawberry in southern and coastal California. And before I started my position in 2016, I was a postdoctoral researcher with Krishna Subbarao in Salinas, working on verticillium wilts of lettuce. Yeah, so today for my talk, I just want to give some basics about the virus and touch a little bit about the host range. So just a brief overview about the group of viruses that INSV belongs to. Don't want to get too bogged down in these details or showing too much graphs like this, but here we have a phylogenetic tree of the genetic sequence of the various viruses that make up the larger group called tospoviruses. And here I just want to point out a couple of things. And the first, you see INSV, Impatiens Necrotic Spot, up here. And so the one thing I want to point out is there are a few other viruses in this larger group that are important to California agriculture specifically, and of course many of these viruses are important pathogens of various hosts. And So, and the second thing to point out is that there are about 5 different groups of these viruses, and one important group contains a few of these economically important viruses to California. So we have INSV here. We have also tomato spotted wilt, which Steve mentioned also. It is has a very wide host range, much wider than the other viruses as far as we know, maybe 900 or more than 1,000 plant species, lettuce being one of them. And it's distributed in many areas worldwide. It's a big economic problem in many other areas worldwide. Another virus, it's kind of newer within the last 9 or 10 years, is tomato necrotic spot virus. Its known hosts include tomato, lettuce, pea, and pepper. And it's notable because in Florida, it's been highly damaging on tomato. And in fact, it's kind of overtaken tomato spotted wilt as the primary virus of this group that is causing problems in Florida. Primarily to tomato and pepper. I don't know if it's actually causing problems in lettuce in the field there. Okay, just another virus is iris yellow spot virus. And unlike the others, this one is in a different group within this TOSPA virus, larger group. And so this one is a big problem on onion, particularly in seed production and in many areas in many Western US states and worldwide. Okay, so yeah, just a little bit more about this group that includes INSV. They can cause similar symptoms, and so they can vary maybe a little bit. And as Steve mentioned, they can be similar with INSV and TSWV, but tomato necrotic spot can also cause similar symptoms. Although for that particular virus, it seems to develop faster and become more severe much more quickly. But the take-home message here is, as Steve stresses, diagnostics is important to identify which virus you have. But of course, as far as we know, again, INSV is the major one that's causing problems on lettuce, and there's been, very few to no reports of, tomato spotted wilt, and I don't think there's any indication that tomato necrotic spot is in California. And another thing about this larger group of TOSPA viruses is that all of them are transmitted by at least one species of Thrips. And Daniel will be going more into this, I imagine, later in his talk. Okay, just another thing I wanted to mention about INSV, and again, don't want to get too into the weeds here, but just a brief statement about what it is as a virus. It's an RNA virus, so it's a ribonucleic acid, and it has 3 different pieces or segments that are wrapped in protein, and then all of that is contained in a lipid envelope that is itself embedded with different proteins. And so this is in contrast to most plant viruses, which have a protein shell and no lipid envelope. And I just wanted to point this out to say kind of how the history about TOSPA viruses in the evolutionary sense is that they believed to have originated as insect viruses, simply viruses that infect insects that later became well adapted to plants. And so that could be an explanation of this different structure here. And another thing about the biology is that's relevant to these 3 segments of DNA is how that affects their population dynamics, because they can come together and kind of recombine. And so say you have this situation of indicating the 3 pieces or segments of RNA with these colored boxes here. So we have one strain in the blue that's contained within the dashed line here. Yeah, it's one particular genetic sequence, and then this other strain has a different sequence indicated by this red color here. So if they happen to have a co-infection of 2 different strains, and we're just talking, hypothetically here in tomato, it is possible for these 2 different strains to what we call reassort, and that it could swap these different the segments from the different strains to form a new strain. And sometimes if it happens between different virus species, there's been a few reports of this happening between different virus species, for example, hypothetically INSV and TSWV. Now, I'm not saying this is happening in California or it's the cause of anything that we're seeing here, but it has been reported in some situations, and it can have biological or practical implications that I'll mention a little bit later. Okay, so just to summarize those few points. So in addition to INSV, there are other tospoviruses that are economically damaging pathogens and can cause similar symptoms. All tospoviruses are transmitted by at least one species of Thrips. Tospoviruses originated as insect viruses that adapted to plants. And so I think others will probably touch on this, but so it's important to think of these viruses as intimately connected with the insects, and they can explain, why they're so damaging because of the host range of the insect vector and other things about its biology. So it's important to keep in mind. And also that RNA segments can be swapped to form new viruses. So this is another reason that regular diagnosis is important. If we're, diagnosing these and monitoring the population of the virus, we can maybe detect some of these. If something happens that creates a new strain, we could detect that early and take some action. So regular diagnosis is important. Okay, so now I'm going to talk about host resistance, and so I want to mention just to give a background slide on some of the concepts, and so I want to contrast two different types of resistance that are two broad types of resistance that you can have in plants. And so the first is complete resistance, and I think many of you would be familiar with this. In this case, you have no disease development under most conditions. And so this is what we have with, spinach or lettuce when it comes to downy mildew resistance. You have a resistance to a particular race. And so that if you had that race of the pathogen, you would not expect to get any disease caused by that race. And so in some cases, for some particular pathogens, you can have a resistance reaction that occurs when this particular pathogen tries to infect that resistant host. So you can have potentially a visible resistance reaction. And this is what we have here on the left. This is a bit exaggerated. This is a lab situation, and not quite sure how often you would see this in the field, but you could have still a kind of, it looks like a disease, but it's a resistant reaction. And so what it's doing is it's preventing the virus from spreading throughout the plant, just in contrast to a susceptible plant over here with pepper affected by tomato spotted wilt. You have systemic disease. It's inoculated in one leaf, and it spreads to the rest of the plant and causes disease symptoms. And so this complete resistance is also called major gene or qualitative. And the second type, which is maybe less familiar, is called partial resistance. And so in this case, you can still get disease on these partially resistant cultivars, but it's just less of it. And so this is also called quantitative, and it's you see it as a reduction in severity, as a portion or area of the plant that is diseased, of a given plant, or incidence. If you look at a field, it's a reduction in the percentage of plants diseased. And this particular incidence or percentage of plants is how the lettuce breeding programs are looking at resistance to INSV. So just want to give a background on resistance to tomato spotted wilt. There are a couple sources of complete resistance to this virus in tomato and pepper. They're different. They kind of have different activity. The tomato gene, as one particular gene, SW5, is interestingly effective against other tospoviruses, including INSV, but the pepper gene TSW is only effective against tomato spotted wilt. Again, this is just to to say that there are just a bit of background on a relative related virus. The one point I wanted to make about this is, of course, like with all resistance, there are strains of the pathogen of tomato spotted wilt virus that have emerged with the ability to overcome these resistance genes. And that's some of these resistance breaking strains were found to have reassorted RNA segments, like I mentioned a few slides ago. So this sort of reassortment, or it can also be called recombination, can have practical implications in the disease in the field. Now for lettuce, there are no resistant cultivars available. I don't believe that there have been any, some of those complete resistance or major gene resistance resistant genes identified in lettuce. However, there are a few cultivars that have been reported to be less susceptible. And so, and not necessarily calling them partial resistance, because they're still working on, this breeding process and developing lines that can be released to seed companies. So just want to, call them less susceptible here. And some of this is, it's ongoing, obviously, work. Ivan Simko at USDA in Salinas published a paper in 2018, identified a few cultivars that were less susceptible than the larger set of germplasm that they looked at. And some other cultivars were identified in a report to the California Leafy Greens research program. And more recently, the breeding efforts led by Kelly Richardson, also at USDA, presented in the spring at the California Leafy Greens Research Conference, identified a few cultivars as well. And so they are, obviously INSV has become a priority for public breeding programs. So both USDA and UC Davis are working on this. Okay. Okay, and then going into host range, just wanted to present a few concepts and talk a little bit about the known host range, but it's Stephanie and Daniel are going to talk a fair amount about this. So I just wanted to introduce some concepts and briefly about what's known in the literature. So when we talk about what does a host mean for a virus, the technical definition is that the virus can infect and replicate in a species, so it can create new virus particles that can go and cause disease in another plant after being vectored. And but as Steve mentioned, you can have different situations. So we're very familiar with a lettuce that shows symptoms. It can be killed by the virus. So that's obviously a host. It's symptomatic, so the plant is visibly affected by the virus. But you can also have a situation in which the plant doesn't show any visual symptoms, or maybe they're really mild or subtle. Particularly in weeds, can be maybe a bit difficult to identify. So we can have both of these situations. And when we talk about a host list, or if we look in the literature and say, oh, there's a list of 300 hosts, for instance, which is the number that's commonly cited for INSV. What does that mean? Does that mean someone found it in the field, in a plant growing in a field, or next to a field in a weed, or did they do something in the lab? And so, if it's a natural host, it occurs without human intervention, or experimental, it was purposely introduced or inoculated in a lab, and that doesn't necessarily mean it was found naturally. So, of course, if it's experimentally verified as a host, there's certainly more potential. You should think of it with this new information, think, well, maybe it's more likely to be affected naturally, but it still was just done in the lab. That's the case for the iris yellow spot virus that I mentioned earlier, that primarily affects onion. It's been shown experimentally to affect some other alliums, but garlic, for instance, it doesn't really cause disease, or is not really found on garlic in the field. Of course, and so when we're thinking about, oh, if we have this list of hosts, what does that mean? These hosts are typically biased toward economically important plant species. And so we care about them more, we look at them more, we study them more. And so this list could be biased in that direction. There's some other factors that can affect our ability to determine the natural host range. For instance, if a host plant is not as favorable as some others, it's not as good of a host. The virus doesn't replicate as much. It can be maybe harder for us to detect that. So, we would need to sample a lot of plants to detect that lower rate of an infection. And so that could be a possibility why we would miss something as a host. Another thing is the distribution. If the plant has never really been in the physical proximity of existing virus epidemic, maybe there wasn't an opportunity for infection, so we just didn't see that infection, even though it is possible for that host to be infected if it was in contact with the virus. Also, another thing, kind of a different aspect to it, if we know something is not a host of the virus, that doesn't mean it's not important for INSV, because this plant can still be a host for thrips. And so as Daniel will probably touch on, how long the thrips can carry the virus for their life. So if they acquire the virus on one host and they move to a non-host of the virus, you still have thrips that can still have the virus. And so that's another thing to consider when we look at these, what is a host or not of the virus. So again, kind of go into these reasons why a particular plant species could be described as a host of INSV. You could have a situation, of course, where we have infection by the virus, INSV, it causes disease symptoms in the field. You could have a natural INSV infection that leads to plant death. Or in a lab experiment, we had some thrips that are carrying INSV, and we forced them to feed on a certain plant species, and that plant later developed disease symptoms. Another situation you could have as a host where INSV is detected in a naturally occurring plant that shows no visible symptoms. Okay, and just on the other hand, want to look at reasons why say if a plant species that is really a host of INSV, but we may not know it. And so it could be that the plant does not show any symptoms, so we haven't tested it. The plant is not economically important, so no one has tested it. Not enough plant samples have been tested to detect that infection, or no plants were near the outbreak, or maybe the suitable thrips vector was not present, so there's no chance of infection. Okay, and just to wrap up here, just wanted to give a brief overview of what kind of crops that can be hosts of INSV. And so there are a range of crops that are grown in California, including those in the desert from a lot of different plant families, including the nightshade, the pea or bean family, of course, the daisy family with lettuce. Other things like cucurbits or things like spinach are also been hosts. Some things that are not known to be hosts include the mustard family and the grass family. So I'm going to leave this up as a summary because I think I'm out of time. So I will move into questions. Do we have any new questions? We have this question from Daniel as a possible explanation for why lettuce increased in INSV on lettuce increased in 2006. So Steve, if you maybe get a chance to answer that one. So question from Victoria, are Brassicaceae and Poaceae, the grass and mustard family, not known to be hosts because they have not been tested? That's a great question. That's probably there's probably an aspect to that. So you may, there may be a case there may be hosts, but as you know, these are large plant families that are widespread, and so we still, I think maybe would need to look into it a little bit more. You know, these are large plant families. You know, they're grown a lot of different places. So, you know, maybe it's not as likely. There's still a chance of that, but it is possible that we don't think they're hosts because they just haven't been tested. What ornamental plants can be INSV hosts? There are, yeah, a lot of them. You know, hundreds. You know, I should. I've gone through that in more detail, but you know, obviously, Impatiens is one of them. You know, there can be a prickly pear cactus, for instance, a lot of different crops, and but I can provide a complete list to you. So sorry, I don't have a better answer to that question. Yeah, I think I should wrap up here with the poll. Give everyone 10 seconds to wrap up the poll. Yes, everyone did well on number 1. Number 2, the correct answer is all of the above. You can have, when we call something a host, it can mean that the virus can be detected in the plant, but without the plant showing any outward symptoms. And number 2, yes, so most people, the correct answer got E. When we think of, if we want to guard against the possibility that something is actually a host, but we don't know it yet, there are a few possibilities, and those are B and C, basically because we haven't looked into that before. Okay, so let's move on to the next speaker, and that's Daniel Hasegawa. So Daniel is a research entomologist with USDA Agricultural Research Service, a crop improvement and protection research unit in Salinas, where he started in 2019. He previously was a postdoctoral researcher with the USDA in South Carolina, working on whiteflies and whitefly transmitted viruses. So Daniel, take it away.
Awesome. Thanks, Alex. And thank you, Steve, and Stephanie, and all the organizers for allowing me to come talk to you today about some of the virus transmission, things that we've learned about Thrips and INSV, some of the host range studies, and just general observations about INSV from the Salinas Valley. Alex and Steve both being trained plant pathologists, I think they can speak in a lot greater confidence about the virology and the symptoms about this particular virus. So I'm really happy and honored to be following up their presentations as an entomologist here at USDA in Salinas. So let's go ahead and just jump into it. So I'm going to try to cover a lot of different topics today. So Thrips and INSV biology, just basics about life cycle and development, some of the transmission requirements, known vectors for INSV. There are 3 Thrips vectors. One of them is really the only concern that we have to date. And then I'll talk about some monitoring strategies, scouting strategies, kind of from a field perspective, patterns of INSV infection, observations that we've seen, and how that ties into scouting and understanding where the pressure, whether it's Thrips or INSV and all of these host reservoirs are coming from. And I think a lot of that dictates the way you scout and training your eyes in terms of looking for some of these potential red flags for INSV. So, and then the last thing is I'll talk about the host range studies that we've been doing here in the Salinas Valley. I won't talk really anything about the work that we've been doing in Arizona or the desert counties in California, but there's some interesting data that have come out there. So Stephanie's going to talk quite a bit about that in her presentation. So our main player here today is the Western flower thrips, and that's Franklin yellow occidentalis. We know they're incredibly small insects, 1 to 2 millimeters in length, and they have a high propensity to occupy plants at extremely high densities, even at the youngest stage of lettuce growth. So A little bit about the life cycle. I mentioned they're quite small. They're a ubiquitous pest. They're called the Western flower thrips for a reason, and that's because they really love and they capitalize on feeding on pollen grains, where they're able to acquire a high protein diet, which complements a lot of the other types of nutrients they're able to acquire from feeding primarily on leaves. So the female can lay hundreds of eggs throughout her lifetime, and really the life cycle from egg to adult can range depending on temperature and different environmental conditions, as well as food nutritional access as well. So 11 to 44 days is the range from eggs all the way to adults. One of the things that I think is important is that they exhibit really, three different types of life stages in terms of where they are within the ecosystem. So the eggs are laid within the leaf. So the female will inject the eggs underneath the leaf surface. Those will develop into a feeding stage, which is the first instar and the second instar. The propupae and the pupal stage are soil dwelling stages. So they're primarily limited to within the top 5 millimeters of that soil layer. About 85% of all of this stage is within the top 5 millimeters of the soil. And that was done by some studies back in the 90s. This stage right here is a non-feeding stage. They are mobile, but they're non-feeding. And then the adults will emerge. The female is on the right, which is a little bit larger than the male. So development requirements. I know someone asked a question in this great list of questions before the meeting. And so I tried to touch on some of that in my presentation too. But in terms of degree days and developmental requirements, really optimal conditions range between 45 to 104 degrees Fahrenheit. Anything below that or above those thresholds, development is drastically reduced. Keep in mind that it's reduced. It's not killing the insect. It's just preventing that efficient development and progression into the next life stage. Ideal conditions is 268 degree days, which amounts to about 11 days to complete the life cycle from egg to adult. And as I mentioned, the host range for thrips is quite large, and that's over 100 different plant species have been described. And in the perspective for Salinas, but also down in the desert, we know that there are a lot of great hosts that will support the reproduction for Western flower thrips. If we talk about Western flower thrips as a vector for tospoviruses, Steve and Alex both brought up the two tospoviruses that are of most significance here on the Central Coast. INSV and tomato spotted wilt virus, they share the same vector, which is Western flower thrips. And then these two bullet points I think are really important, and they're going to be in your poll questions as well, so pay attention to these. So some tricky things about the life cycle, but really important to thinking about transmission is that the virus must be acquired during this larval stage in order for them to transmit as the adult. And I'm going to go into some details about that and some great pictures to really try to, burn this idea and these concepts into your mind about how transmission takes place, because it's quite different from aphids and whiteflies. So, the larval stage has to acquire the virus. And so what that means is that really the female has to lay her eggs into an infected plant. So this is a mobile stage, but they're not that mobile. And so if an egg is laid into an infected plant, the emergence of this first instar is the most optimal for them to acquire the virus. And then they have to complete their entire life cycle. And then it's only the adult stage that can transmit the virus. So the larvae cannot transmit the virus. They can only acquire it. And only the adults can transmit the virus. When the female lays her eggs again, that egg does not have the virus. And so they have to reacquire it every single generation as a first or second instar. Okay. Let's see. So other things that are important in terms of thinking about transmission, INSV cannot be transmitted by contaminated clothing or farm equipment, airborne particles. If we start thinking about COVID, INSV cannot be transmitted that way either. It has to be transmitted by the insect in that manner that I just described. Other insect vectors, aphids and whiteflies cannot transmit the virus. And what I think is just really important from a vector entomology standpoint is that a lot of the viruses that you deal with down in the desert affecting melon production, CCYV, CYSDV, a lot of these are acquired and transmitted by the adults. So the adults can feed on an infected plant, acquire the pathogen, and then transmit it as an adult again. And that's quite different from the way we think about Thrips transmission of INSV. The other challenge is that the host range, as Alex touched on really, wonderfully, and as well as Steve, is this idea of the host range. So over 100 different ornamentals, food crops, weeds have been described as a host for this particular virus. This is that little picture I was telling you about. So I'll walk you through this if I wasn't clear enough about the transmission in the last slide. Hopefully this will help. So we have two different scenarios. We have an uninfected lettuce plant, and we have an INSV infected plant, and are the two examples. So these are all kind of no-brainers, these first two scenarios. If an adult feeds on an uninfected plant, you're not going to get transmission into another plant. I just saw a question pop up, can knives transmit it? No, the mechanical transmission is not going to be an issue in the field. Second scenario here, if the female lays her egg into an uninfected plant and then completes their life cycle on that uninfected plant, you're not going to get transmission to another host. This is the tricky part, right? So if an adult feeds on an infected plant, you still do not get transmission, right? And that's different from a lot of the aphid and whitefly-borne pathogens. And it's only when you get the female that lays her egg on an infected plant to allow the larvae to acquire the virus and then complete their life cycle as an adult. And only under those circumstances do you get transmission. Okay. There's another question from Armando about how far thrips can travel, and that's something we'll touch on very briefly in a little bit. So great question. Two types of infections is the way you think about it. If we have an infected plant here and there's more infected plants in the field here, the first one is if you have an incoming population of viriliferous thrips, as you call them, so adults that are carrying the virus and are able to transmit it, that's your primary infection coming outside of the field, introduction into your field. If you get reproduction, egg laying, and then persistence within that field and then further spread, that's what we call secondary infection. And so this is just kind of terminology that I try to use when I talk to our industry here, because when you're looking at a field, sometimes it's difficult to understand where the source is and how that virus is propagating throughout the entire system. And so primary infection, secondary infection are very general ways that we think about modes of transmission as well. So known vectors for INSV, there are 3 described vectors that we know of. And so really this was this one on the left here is the Western flower thrips is our major concern. And that was, this work was published by Steve Koike and some other great scientists at Davis, but they did a survey back in 2008, 2009 and showed that 84 to 98% of the species collected from lettuce were Western flower thrips. The rest, most of the other ones were onion thrips, which is not a vector for INSV. European flower thrips, tobacco thrips are also described hosts, but we don't have any evidence that these insects are here in California or Arizona. This is some of the work that we're starting to do, is doing some massive population studies to really try to understand if there has been a shift or an introduction to some of these other vectors that might be contributing to the reemergence of this pathogen and some of the other really great epidemiological studies that we have. So this is getting into flight. So this is also a poll question, so pay attention to this. So thrips in general are poor flyers. You can see that this is a male. They have really, the wings are not considered true wings. They're really kind of bristles. And so they rely heavily on wind for dispersal. Distance is quite hard to imagine, to estimate. So unfortunately, we don't have a great answer for that. But if you think about migration, a lot of migration is triggered by removal of the host crop. So whether that's harvesting or disking of lettuce fields where you have established thrips populations, whether it's a neighboring field that is being harvested, whether you have a weed abatement system occurring, these are all triggers that can help facilitate thrips migration, right? And that makes sense. You remove the food source for an insect population, they're going to pick up and move to something else. Okay. And so when we think about scouting efforts, you just want to be aware of the wind direction, surrounding landscapes, different farming activities that are around you, and particularly focus your scouting efforts on where some of those higher risk situations might be occurring. So some studies that we've done in the Salinas Valley last year is we just looked at the height of thrips movement. And I think many of you know that we get some very strong winds coming in, offshore breezes from the Monterey Bay every afternoon, sometimes up to 20 miles per hour. And so being windborne and relying heavily on the wind, this isn't all that surprising to see that thrips populations are being captured in that wind column up to 10 feet and probably even higher. And so again, thinking about where you are with respect to the wind and your neighbors and how far other people are, upwind and downwind from you, I think those are all important in terms of facilitating how you conduct your scouting efforts. And so I just covered that first section. This scouting strategy is going to be, really what I just talked about, but scouting edges, corners, transitions, and really thinking about these periods of high activity where you might be facilitating the migration of thrips populations. This is just a picture showing that we have a harvest here, another crop of lettuce that's planted that's about midway through, and a newly planted field right here. And so you can imagine the migration of thrips between these fields, but also the challenges once you introduce the virus, how is that virus going to move based on the movement of thrips populations. So in 2020, we did the study. I'm not going to spend too much time on this, but we have our wind coming in from the north. In a field that was planted from February to April, we had a high gradient effect of the virus. So again, reinforcing that idea of scouting edges and corners and some of your transition periods, we saw the virus move to these edges, presumably because of the wind that was moving down here. We had another source on this side over here that introduced the virus to fields 2, 4, and 6. The reason why I don't think we saw a lot of incidents that happened in a lot of these fields is because we were still pretty early in the season. And again, as Steve and Alex mentioned, that the dissemination of the virus is strictly driven by thrips populations, and you see the thrips populations are still quite low when we have introduction of the virus. And so I think that is helping this particular system, is that you're reducing the chances for the virus to spread because you don't have the vector in the system quite as high as you have it, certainly later on in the season. So this was a question that came up earlier. Symptoms in lettuce, I have approximately 10 to 14 days, but again, that is strictly dependent on, temperature and environmental conditions, certainly varieties. And so, Steve's point of a couple days, I would agree with that, definitely under optimal conditions. In larger plants, you can see symptom development take quite a lot longer, just because there's a lot more tissue, and sometimes it may not be obvious. So this plant here on the left is infected, and you're starting to see the onset of disease of symptoms coming on here, but it's not obvious just looking at the plant above ground here. You really have to peel it back. And so it really is, this sliding window depending on where you are, the crop stage, the variety, temperature, all of that. So and I think that was one of your poll questions too, so keep that in mind. And I'm just going to show some pictures from observations from the Salinas Valley. Steve did an amazing job talking about symptomology, and I'm not going to talk symptomology here, but I'm really just going to show you pictures of how we've see this virus in its worst form in the Salinas Valley, and hopefully having a repeated amount of images of the pathology side of it, but from the field side, will help you become better scouters to know what you're looking for, and whether it's diagnosing it as INSV or at the minimum being concerned and then sending it in for testing to Steve or another lab to assist you in the confidence there. So 2020 This was another field where the symptoms were quite limited to the midrib within the heart of the plant. And so again, it can look a little bit different. These are, you can see a great discrepancy in plants that are a little more established, whereas other ones are infected quite early. And you see that severe stunting that's caused by the inoculation from the Thrips vector. Same thing in 2021. That are established, whereas a lot of these other ones have been inoculated quite early in the crop cycle. So in 2019, I really enjoy these pictures. It's unfortunate for this field, but the inoculation came extremely early at the pre-thinning stage, where you're already seeing strong necrotic symptoms on these young plants. So just to document the severity and the spread of it over a 9-week period, this is some work the former entomology farm advisor Alejandro Del Pozo. Again, in 2019, we collaborated with JP Dondore-Ardias at CSUMB, who's a plant pathologist. I'm not going to talk much about the sclerotinia work. We have that data here, but really the important piece here is that, you know, following the harvest, we did pre-evaluations and post-evaluations, and we saw that there were still, you know, about to 30% of the field was harvested, even though there was infection within those plants. And so what I think ultimately this is telling us is that management of Thrips vectors and INSV, your best chances are really early in the crop cycle when you have better coverage, and you know, hoping that, you know, those later infections are not going to affect affect the symptoms to the point that you lose the market quality and the harvest potential for those crops. So that's also in your question, in your poll question too. So one of the things that we do here in the Salinas Valley is we monitor Thrips populations on a weekly basis. And so we have 21 cards set up. All right, so Thrips populations, we generally see them increasing as we progress through the season. And that correlates with INSV incidence. Just to put that into perspective, those studies that we did in 2019 was in that planting slot right as Thrips populations were increasing. If we talk about INSV incidence, in 2020, we see, let me just move this out. So yeah, so we generally see the highest incidence from Salinas to Gonzales, and that was in 2020, but then also seen in 2021. And this has been a great reporting system led by the Grower Shipper Association, and that's the INSV Pythium Task Force that I think many of you are aware of. So far to date, 211 fields have reported incidents greater than 1%. We continue to monitor new fields and those that have, you know, some of the higher percentages if those growers are willing to disclose their locations. So that was the top two sections that were covered. Please let me know if you have any questions. The last part I'm going to cover is the strategy to identify the host range in the Salinas Valley. And so if we think about the end of the 2019 season and then the 2020 season, we had two strategic sampling efforts during the lettuce-free period to really address whether some of these weeds are playing a role as bridge crops for the virus between lettuce plantings. So spring, summer were also covered. I won't talk about those in individual details, but mostly as a whole to talk about the big picture. Our primary method of detection is using those serological tests that Steve mentioned previously. We also run genetic tests to do a form of validation, just to be sure that if a sample has the virus, then it actually that we're absolutely confident in what we're calling is a host for the virus. So 4 sampling periods were conducted over 4 locations. In general, our north and west locations experienced moderate to high levels in 2019. In the east, between Chular and Gonzales, we had our highest levels of INSV. And then we had some pretty low levels in Soledad And so we wanted to include that as a southernmost location to assess whether we were finding the virus in the weeds that were collected in this region as well. We also selected these based on some of the geographical unique features in these areas. So urban areas, industrial areas, riparian habitats, foothills, etc. So we tried to address a lot through these strategic sampling periods. In summary, we've tested over 3,000 plants. That number is probably closer to 4,000 now, and that ranges over 73 species, most of them being weeds and vegetable crops and some other things as well. So the strategy for ranking which hosts are potentially the best hosts as reservoirs for INSV, again, we've had numerous samples that tested positive, but the goal for this was to really boil it down and to say these are the top 10 hosts that we think are serving as reservoirs for the virus. And so this ranking system that we developed is dependent on this number, which is reflective of the virus titer within that host plant. And then we multiply that by the percent positivity value as well. I have lettuce and chickweed up here that both have asterisks on them, and that's alluding to Steve's point about this idea of symptomatic host plants. So lettuce and chickweed are both plants that show very strong symptoms of the virus, whereas a lot of these others do not show symptoms. And so it can be challenging to really go out and sample a lot of these different hosts, especially when you're seeing herbicide damage, old age of some of these weeds. And so being able to diagnose something and to say it's symptomatic of the virus is especially challenging with a lot of these other weeds. And so we rely heavily on these serological tests and the validation that we do to develop these proxies for determining which ones are our best hosts. And so in combination with this ranking system, we also think about the seasonality and the abundance of the weeds, which I caught when I was able to jump back on. I saw Stephanie talking a little bit about that as well. So many of these are considered annual plants, annual species, but here on the Central Coast, a lot of them behave unlike annuals, and their presence is throughout the entire year. And so we think that is contributing to the potential development and establishment of total virus inoculum in the Salinas Valley. And so we've really tried to focus on these top 10 hosts here. These are just pictures. I can share it with your industry as well, but this is what we circulate and what we talk about when we approach the Ag Commissioner's office, when we talk to third parties, and we really focus on a lot of these weeds because so far from the work that we've done, these are the ones that are consistently positive for the virus. When we go back to our some of the data that I was mentioning earlier, focusing on the top 10 list, over 3,000 samples tested, and overall we saw a 13% positivity rate. If we look at limit that to our top 10 list, we see that percentage jump up to 20%. And so that's also another form of validation that our top 10 list is actually those of the most significance. So numbers here don't mean much, but when you look at it graphically, it's quite impressive. And so you look at the winter sampling period of 2019 as well as 2020, and we see those numbers for our top 10 list be quite high in terms of percentages of plants that we're finding that are detecting positive for the virus. And so pretty good evidence that they are serving as bridge crops for the virus between subsequent seasons of lettuce. If we look at that same data organized by location, I mentioned part of the strategy was looking at regions where we saw different levels of incidence within lettuce and then sampling the weeds around those areas. And so if we just jump to the graphical part of that as well, the south where we saw the lowest amount of INSV and lettuce, we're also finding very low titers and levels of virus in the surrounding weed habitats as well. Over on the east side where we saw the highest amount of INSV in 2019, 2020, and we're seeing the highest percentage of weeds and non-lettuce crops that are testing positive for the virus. So again, another layer of validation that's supporting the role of some of these alternative reservoirs for INSV. In addition to just managing weeds in general, I think our growers and our industry are really good at managing what they can control, but some of the issues that are emerging are where these weeds are, and where they are may not be within the control of one particular grower. And so there has to be a systems-wide approach to management. And I think a lot of the data that we're generating here is starting to facilitate those conversations of getting other growers that may not be interested in lettuce, but improving management for the sake of the economics and really just the production of lettuce in our region. So in summary, we've tested several foothill riparian habitat species. We haven't seen any indication that we haven't found the virus from any of those, and then a lot of other ones that are of industry value to our area, including sweet alyssum, which is used as insectary plants. Some cover crops as well as ice plant, which we know is a great host for thrips. We haven't seen it test positive for the virus, and that's used for a number of things, including erosion management on some of the banks throughout the valley here. We've tested a number of other crops, some of which have come back positive, others have not. So just to wrap up, many of you have seen this. It's our IPM model that we're developing for the management of both INSV, but also thrips, and what we can do to try to improve the management for both the pathogen and the insect vector individually, but also thinking of it as an entire system as a whole, because I think it's really when these two come together which is where management is really needed the most. And so integrating different facets of management from a systems-wide perspective, both to lettuce management as well as non-lettuce growers, I think is going to be critical as we move forward in the Salinas Valley. So a lot of great people to thank, including the people that I've been working with down in the desert as well. You guys have a great team down there with Stephanie and John Bindu, and then now with Akurba and Alex coming from the UC side. I think you're well positioned to really understand this pathogen and its potential emergence in the desert region down there. So, but I'm up here on the Central Coast. Of course, if anyone has questions, feel free to reach out to me or reach out to John or someone else. They all know where to find me. So
you have a few minutes for questions. And so the first question from Philip is there a benefit to the thrips when they carry the virus? For example, a longer lifespan or increased egg production?
There have been some studies, not done with INSV, but they have done quite a few studies with tomato spotted wilt virus, which is that other Tosco virus that Steve and Alex talked about as well. And there are differences in fitness. It can affect egg production. One of the more important things I think that has been interesting that comes out of the spotted wilt literature is that in some circumstances, when a plant is infected by the virus, that plant is the changes in the volatiles in the plant makes that plant a little more attractive to thrips vectors. And that makes sense from a biology standpoint, right? The virus and its transmission and its ability to replicate is strictly dependent on the transmission of the insect vector. It's this theory that runs through academia called the vector manipulation hypothesis. And so how does an infected thrips vector change in response to acquisition of the virus? So there is a lot of information on that. For spotted wilt, we don't know anything about INSV.
Okay, thank you. Are there any questions live in person?
So how long does it take for the larva to become infected when they start feeding on a plant? Does it become immediately, or do they need to be feeding for a certain period of time to become infected?
Yeah, so great question. So the question was, how long does it take? Do the larvae have to feed? And what is that window for a virus acquisition? And For transmission from the adult side, they are most efficient and they have the highest virus titers within them when that larvae feeds immediately after it comes out from the egg stage. But we know that acquisition can happen within hours. They can acquire the virus. One thing that I don't know if I mentioned it, or maybe I mentioned it when I cut out, but this virus also replicates within the Thrips vector. So it's replicating in the plant and it's also replicating in the Thrips vector. But the sooner they can acquire it, in as little as, several hours, that insect will be hot for the virus as an adult and will be able to transmit it. And transmission, we know, can occur within several minutes. So once the adult has the virus acquired from the larval stage, they go onto a plant, feed for 5 minutes, and you get transmission. And that idea of the efficiency of transmission is not all that different for whiteflies or some of these other insect vectors as well, where we see the efficiency being quite large.
Yeah, great question. And a follow up, a couple of questions online related to that.
Sure.
Is Thrips a persistent virus vector, and how long do they carry it, the adults?
Yeah, so the adults, the adults will carry the virus for the remainder of their lifespan, and they'll continue to transmit the virus throughout their lifespan as an adult, but the efficiency will start going down as the adult ages. So, yeah.
Okay, and therefore, because it's replicating, it is a persistent virus vector.
That's correct.
Yeah. Okay. Maybe one more question here. So Jeanette is wondering, similar to what's been done on the coast, are there ongoing projects to establish spatiotemporal dynamics of INSV infection, specifically for desert lettuce? And I think Jeanette may be asking about that tracking study where you showed the map in a field. If I'm got that right.
So tracking the virus, that's really been led by Grower Shipper Association of Central California, and the reporting for that has been really a wonderful thing that Mary Ziske and Chris Valdez have really facilitated. There are issues and concerns with privacy, but the locations of those areas are never disclosed in all of our meetings. It's not something that I am spearheading, but I've helped in really the launching of that, and I think it's a great system. I don't know if there are plans down there in the desert for it.
Okay. Yeah, so we closed out the poll when you got disconnected. So I think it's you can see the responses now. Would you mind see if there's anyone need to go over?
Yeah, so which of the following is true for question 1, and all of the above is correct. So 79% of you got it. Well, technically all of you are correct because all of it is true. So INSV cannot be transmitted by contaminated clothing or farm equipment, cannot be transmitted by windborne virus particles, cannot be transmitted by other vectors. D is true. It can only be transmitted by thrips, but all of that is actually true. So technically the right answer is E. Which of the following is true for question number 2? And it's the same thing, all of the above. So thrips larvae cannot transmit INSV. Thrips larvae can acquire the virus, but they cannot transmit it. B, only adult thrips can transmit INSV. That is correct. C, INSV must be acquired as larvae in order to transmit the virus as adults. That is true, but technically all of them are true. So D is the right answer there. And the theme to all of my questions, I think, is that all of the above is the right answer for all of them. So technically nobody's wrong because at least one of the answers is right. So do you want me to go through all of these, Alex, or?
Yeah, I think everyone did well on number 3, 4, and 5. So
yeah, looks good. Okay.
Okay, yeah. Okay, so thank you very much, Daniel, for an excellent presentation. And Dr. Stephanie Slinski is the Associate Director of Research at the Yuma Center
Back up. Oh, sorry.
Is the Director of Research at the Yuma Center of Excellence for Desert Ag. She's a plant pathologist with broad research background working in fungal and bacterial diseases of vegetable crops, ornamentals, conifers, and citrus. Dr. Slinski has experience working with researchers and with the agricultural industry to develop solutions to the pressing production needs. Before coming to YCEDA in 2018, Dr. Slinski worked with researchers and the Florida citrus industry to develop research and manage citrus research funding. So go ahead, Stephanie.
So to start off with, we started seeing INSV in early March of this year in Arizona. And I went out with a PCA and took a look around the Tacna area and got a good idea of what was showing up. And, we talked about, what was going on, where did this come from, and all these unknowns that are still unknowns. But we've looked at this one particular field, which was a pretty severely affected by the virus. And around this field was alfalfa. So that's when the question came up about whether alfalfa was a host, because you see a lot of thrips on alfalfa. What's going to be the impact of this on the spread of the virus in the future? So, I took a look around. If you look at this close-up I have here, that shows a thrips nymph on the leaf. And so, if alfalfa is a host, we have a lot of thrips on it. It could be a really big problem. So what I did was I took out one of our lab technicians, and we did a pretty extensive sampling. And we sent samples of alfalfa to Daniel Hasegawa, and he was nice enough to test a number of these samples for us. And we found that the alfalfa was all negative. This isn't a confirmation that alfalfa is not a host. It's possible that we just didn't select any infected plants. But for the most part, we weren't able to get it. We weren't able to detect it. So this led to other questions about what else is out there that could be a host and be a way for the virus to live over the summer and infect that fall lettuce planting. And so this turned into a survey to help answer this question. So are there weeds that can act, or are there other crops that can act as a green bridge between lettuce growing seasons? And the weeds that are around, they have to be a host of the virus, but they also have to be a good host for the thrips as well. So what happened was we started collecting weeds weekly from the Tacna area and also in some other areas around Yuma. And I alternated with Dr. Palumbo to do this. And we sent samples to Daniel Hasegawa in Salinas for analysis. And we've been doing this since early April. And this is kind of a non sequitur, but I wanted to add that we don't know how INSV was introduced into Arizona. We probably will never know. This survey is not going to answer that question. But it will give us an idea of where it might come from, where it might have come from if you see it early in the season this year. So it might have come from other hosts in the environment. So in early April, the lettuce crop was still in the ground. There were lots of weeds around. This is showing the edge of the alfalfa field. And there's a wheat field next to it with a lot of goosefoot. So, you know, there's a lot of green plants around for the thrips and also for the virus. So I collected weeds from all of these areas, including from around the lettuce plants in the field. And we did get a number of positive samples. The positivity rate was pretty low, but we did get a number of positives. We collected a pretty good amount of samples too. So you can see that, you know, some of the common weeds that you see around here had the virus. So especially the nettle-leaf goosefoot, the sow thistle, purslane, cheeseweed, those are some pretty big hosts. The clovers were the ones that I collected right from the lettuce field. They were right up against infected lettuce. So that's why they're likely why it's 100% detection in those weeds. And so in May, we collected a number of samples again, and this is our 4-week numbers. And again, we consistently saw infection in a lot of those common weeds: cheeseweed, lamb's quarters, nettle-leaf goosefoot, sow thistle. So we continued to see them through May. And in June, the alfalfa, well, you know, the alfalfa was mowed several times, but on this one day, I saw that the alfalfa was mowed. The wheat was still on the ground, and it looked like the goosefoot was kind of getting burned down. You know, it's getting towards the end of the season for the goosefoot, but there was still some green goosefoot in there, and but the edges of the alfalfa field were pretty well cleaned up. So we're seeing less weeds in early June, and we're also seeing some of the crops are getting harvested. Mid-June, we saw that the alfalfa field was harvested. There was still a little bit of goosefoot there, but the alfalfa was regrowing, and there was a lot of weeds in there. So, you know, there were still weed hosts around to carry over the thrips and the virus. But we did start seeing fewer samples positive. So still cheeseweed, sow thistle, goosefoot, but we were getting fewer weed samples in a lot of cases, and also fewer positives. And on that last sampling date, we got no positives. And that trend continued in July, and we just recently got samples or results from Daniel, and they were all negative for the 2 sampling dates after this. So our samples are all negative, but this isn't necessarily a confirmation that it wouldn't be possible to find a weed sample that was positive. We would have to sample pretty extensively, but it does seem like there's less of the virus in the environment. Most recently, or towards the end of July, things are pretty burned down because of the heat. But last time I was out, I was starting to see more weeds, including some of these hosts, like lots of purslane. The fields are being prepared for planting. You're seeing a lot more purslane. This is the corner. The middle picture is the corner of the alfalfa field, and there's a lot of amaranth in there, and it doesn't seem like amaranth is a host. Palmer amaranth was just, that was a host that really was doing, or sorry, not a host, a weed that was really growing well over the summer, but it was never positive, and it's not reported as a host. So that's good news. But in late July, I was starting to see some nettle-leaf goosefoot regrowing. So, we are starting to see more of those good hosts coming back. Here is a summary of all of the samples that we did through mid-July. And Nettleleaf goosefoot, cheeseweed, south thistle, lamb's quarter, purslane. Those seem to be pretty good hosts. You see the positivity rate is pretty low, but we didn't see a lot of INSV in the area, so it's probably not really high in the Thrips population. So the chance of, or it's not spreading very fast. So these all seem to be good hosts based on some of the work that Daniel's done, and we might expect to see higher positivity rates as time goes by and the virus becomes more common. So I already mentioned there were some weeds that I collected that were adjacent to the infected lettuce. Those are why these are at 100%. It's not because these are better hosts. It's just because they had more chance of getting the virus. And you know, none of these were positive. So does that really mean these are non-hosts? And that Alex had a good slide talking about this. It's really about how much you sample. It's about when you sample. So if you sample really early, the plant might not be systemically infected. There's, you know, could have escaped infection. It's about what proportion of the thrips have the virus. And of course, the seasonality. So some of these, we didn't collect a lot of samples like shepherd's purse. I got one sample. You don't see a lot of that over the summer. It has been reported as a host. So yes, the one sample I took was negative, but this is reported as a host. So, and also the London rocket, that's reported as a host. So, you know, I would say something like, you know, here we had 89 samples of amaranth. It's likely not a host, but we still need to there's more studies that are necessary. You have to do transmission studies to really confirm whether it's a host or not. It does look like, you know, alfalfa, the 161 samples we took, they were all negative. I still don't take that as a confirmation. I would want to see a transmission study to confirm that. So the next steps to take with this is to confirm, we really want to confirm the importance of these weed hosts over the summer. So if we want to know for sure if you really have to do intensive weed management over the summer, like we want to confirm that the weeds are important over the summer, so you know you need to manage them. So what we want to do is we want to go in and do a real intensive sampling of weeds in the early season. So we want to, well, fields that when you're preparing your fields or when you're starting to plant, we want to go out there and just collect, say, 1,500 samples and see if we can detect INSV. So that'll give you a good idea of, you know, whether you have to wait for INSV to be introduced again or if it's already in the environment. We also want to track the incidence over the season, so coordinate some weed sampling with the Thrips monitoring. And I put in a proposal to the Arizona Iceberg Lettuce Research Council to do this, and that's with John Palumbo, and hopefully we'll be able to do this project. So there's some good gaps that the INSV Pythium Task Force put together, and these are pretty useful for the desert. It's worth refining them for the desert. But one of their recommendations is to aggressively manage weed hosts. So that's including in the border areas, and so that's a good, you might see this as a poll question. So, you know, it's good to take note of this, and it's good for you to think about when you're preparing for next season. So thank you all for listening. And I wanted to say that I want to acknowledge the donors to YCEDA. Because of them, I was able to just go out in the field and collect all these samples and not have to wait for funding. And Daniel Hasegawa was very helpful in helping us collect samples, helping us test samples. It was a really great team. John Palumbo collected samples as well, and I'll take any questions. Okay, Alex, can you tell me how everyone did on the poll?
Okay, yeah, so number one, 66% selected A, unknown, for how was INSV introduced to Arizona. So do you want to address that?
I can't see the poll question.
Okay, yeah, well, I'm saying only 66% got A.
That was the one that was, how was it introduced? And
Yes.
we don't know. It's impossible to tell at this point.
Yeah, yeah. Number two, what weeds have been found to be hosts of INSV in Yuma? Almost everyone got that, E, all of the above. And number three, what reasonable method may help reduce the risk of INSV? 73% selected A, control weeds on field edges. And others, B and E, also some people selected.
Okay, but most people got that one, right?
Yeah, yeah.
Okay.
Marco Peña is an assistant in extension at the University of Arizona, Yuma Agriculture Center. He started his career at the University of Arizona in 1992 at the Department of Entomology. He's currently working with the Arizona Vegetable Integrated Pest Management Team in the weed science area. Marco has been assigned to coordinate the publication of the Arizona Vegetable IPM Updates newsletter for the past 12 years. Mr. Peña has a BS in agronomy from the University of Baja California and MS in agricultural education from the University of Arizona.
Well, I want to thank you for the opportunity for talking to you. And I wanted to mention, I wanted to thank Larry Tickets for his insight in the seasonality of the weeds. And he's a maestro. And there's the weeds right there. The top 10, like Daniel mentioned, we got them right there. Purslane, Malva, Mustard, Shepherd's Spurs. We got South Thistle on the left bottom, Lamb's Quarter, Goosefoot, Mare's Tail, and Bindweed. So we got and we want to talk about the seasonal, the way they grow, the growth habits, and we got some summer annuals. We got biennial weeds in there, and perennial. And in this conversation, could we keep in mind those weeds to maybe look at the implications of the life cycle to have a possible host-free period. So let's check them out. As you know, the winter annual germinate in the fall. They spend the winter. They produce flowers and seeds, and in the summer, higher temperatures, they die. Supposedly, that's a true winter annual. And those are the ones in PCA study guide for Arizona. But I highlighted right there common lamb's quarters and goosefoot for some reason. Some of the winter annual weeds can survive way into the summer. If they're not caught, in other words, if they're not in an alfalfa field or something, they're caught, they're in a shaded area and they have access to water, they can survive. They survive way into the summer. Like for example, we got right here, South Thistle in October, South Thistle in June. We got goosefoot in October and in June. It almost looks like a different species if you look at it, and but it is the same goosefoot. It was one of the top 10 weeds that was positive, right? That was last week, August 5, 2021. That's snail-leaf goosefoot. And actually, this morning I stepped in a field and look, it doesn't look like a winter. I don't feel like it's winter, but we had a winter annual growing pretty close field in a field. I'm not going to name what this field is, but it's right here in the area. So what I wanted to mention with this weed is that the name of winter annual, summer annual, it's different if you apply it in like in the San Joaquin Valley. I was looking at the San Joaquin Valley map. In some areas, this is considered a winter, a summer annual. Here is a winter annual, but over there it's a summer annual. Well, that's new to me, including the very close related plant, common lamb scoter. This Chenopodium morale, Chenopodium album lamb scoters is also considered summer annual over there. And where I'm going is that IPM is local. We all have heard John Palumbo says that IPM has to be applied to our area, to our low desert area. And of course, sweet seasonality is local. Shepherd's purse is another case, and it's a winter annual here, but it's commonly found in some of those counties that were mentioned in the Salinas Valley. And of course, I mentioned that leaf goose food. Let's go to summer annuals. But they germinate in the spring, and the cycle is, as we all know, this is a review for all of us. They flower, and then first frost or in the fall, they die. Supposedly, that's a true summer annual. But I put the purslane right there in the corner because I wanted to mention something about common purslane. We have seen it in our trials, on we science trials here at the station in December and germinating in January, competing with our lettuce plantings. And so the point is that some summer annuals weeds can survive way into the winter, especially if we have mild winters or there's some hot spots and cold spots, and there's a possibility of them surviving in crowns, and specifically purslane cut a little section in roots from the nodes. And it's reported that this weed germinates in 24 hours from August, September, October. But and it extends November, 48 hours, and it extends the time to germination. But it still germinates in fairly cold weather. The biennial life cycle. Those are the weeds that they say they germinate in a spring and they continue growing. Sometimes the first year they don't produce. They just hold on in the dormancy and at roseted form. They don't produce seed until the second year. Temperatures are favorable, and then they flower and then produce the seed. It could be one flowering cycle, two flowering cycles. This is interesting because Malva was reported as one of the INSV hosts, and yellow sweet clover. But in this case, this picture of a white sweet clover. And I neglected to put right there Marysdale. The Marysdale supposedly acts in Arizona as a biennial too. So it's going to be available for during the whole summer. We're going to have it. It's out there right now all over the place. And it's there's Malva in October and Malva in June. If you look at the plant here on the right, do you think that can provide some green material, something like a bridge from one season to the other? I mean, it's a pretty tiny little plant. It does not very healthy, but I don't know about the if Western flower Thrips, Frankliniella species will breed on it or not, but there is the material. That's my point right now, is the seasonality. I mean, Dr. Palumbo is going to explain to us this, if Frankliniella species would produce the larvae or not. But at this point, that's what we're looking at. Another one that I mentioned is another biennial, which means that it's going to be one cycle or two seasons there together. It may overlap, and it's out there somewhere, right? Like this. I mean, this is annual, but this is a winter annual, and it's here in the summer. Anyways, let's go. That's Marysdale in October. The perennial life cycle, well, it's just a review. We know that First year, they just establish. The following year, there's plenty of roots and very complicated to control them. They're very well established. Those are the ones that are in our PCA study guide for Arizona. This field bindweed right here. And they have different organs that reproduce. They're very they're very, very easy to reproduce by stolons, by rhizomes, tubers. Like there was one that on the list that we have here in Yuma. It's a hog potato. It has tubers on there, and you can spray a PPO on top, kill it or hoe it, but it's going to come out from the tuber. It's going to shoot another plant. If you got stolons, are the succession of nodes on the surface, or rhizomes, the same thing, succession of nodes but underground. So their seasonality is extended that way, and of course by seeds. There's like a planning chart right there, slide. The more planning you do, the impact of the problem will be reduced. That's what I intended to say with that chart. So what kind of steps are we going to take? Similar critical steps that agencies take, like governmental agencies, is like this. Arizona prohibited noxious weeds. We're trying to avoid the establishing of one weed, or if you go to prevent the establishing of a disease, there's a situation like our friends in California are doing for the lettuce mosaic virus, a host-free period from December 7 to 21. Avoid the hosts for those weeds. And those are a couple more examples. I know I don't have much time. So we could look at the germination calendar for broadleaf weeds in Arizona. If we know when it is likely that they're going to be around germinating, it's good to have that data. Talking about germination, the time to germination, very tickets, and you probably remember a grower, Dennis Montepenny here. They did a trial. They put 7 different weed species in tea bags, put them on the ground before the flood irrigation on lettuce fields. And they did many locations, and then they came out with this information. Some of these are positive or could be hosts of INSV. So, like for example, as I mentioned, for instance, purslane 24 hours in August, September, October, and 48 hours in November for germination, December 96, 168 hours in January. And if you think about it, I mean, purslane is germinating in January. How much longer till we'll go back to the other season, to the spring? So practically, it's going to be around all year. And if it's a good host, there's a possibility right there of carrying from one season to the other this virus. So these events happen all year round. So I wanted to put that in kind of like a cycle. As you can see, that's the year around. Every section is a month. And you can see the weeds that are possible source of infection. And on the left side, you can see it's a very populated area right there. A lot of weeds, and from October to March. So we see the possibility of creating, at least in our own operations, like a host-free period. If you're aggressive, I know everybody's talking about sanitation. It's what's showing right there. If we go aggressive on purslane, if we go aggressive on mare's tail, and we have mallow, of course, that's another biennial. This and the escapes for the goose food, that would definitely help us mitigate the problem of INSV transmission from one season to the other. For purslane, I know you guys are the experts on the herbicides. Those are for summer crops. We got tools for them. We got tools for Caniza canadiensis, horseweed, or the flea vein. Because I detected that was positive too, flea vein. So those are the tools that we could use. So with that, I just want to mention that can weeds provide green bridge harboring INSV between lettuce seasons, yes or no? With mallow as a biennial, covering the whole summer like we saw. Purslane, Marysdale with the goosefoot escapes, lamb's quarters. We believe this is possible. Yes, that's what we believe. And again, whatever we already covered, that's what we wanted to cover. And with that, thank you for your attention.
All right, thank you very much, Marco, for that excellent presentation. That's a nice figure you showed there, showing the the different periods that the weeds are active. So I don't have any questions in the online here. Are there any questions in the room there?
They're all experts here.
Yeah.
Thank you.
Okay, thank you. So wait. So let's give everyone 10 seconds to finish up the poll.
Oh yeah, on I mentioned it on the question. The first question was B and C. The answer: cheeseweed, lamb's quarters, south thistle, and shepherd's spurs positives. And the second question was December and January, B. And the third question was weeds can actually be a source of INSV infection. And in this case, the answer was false, a true false question. Thank you.
So John Palumbo is a professor and extension specialist in the Department of Entomology at the University of Arizona. He received a BS and MS in entomology from the University of Arizona and his PhD in entomology from Oklahoma State University. He has been a faculty member at the Yuma Ag Center for 31 years, where he has statewide responsibilities for applied vegetable and outreach activities related to insect pests of leafy vegetables and melon crops. His research activities involve addressing immediate problems in local and regional vegetable crops, as well as working on more basic long-term IPM approaches. As an extension specialist, he strives to develop science-based outreach programs that emphasize the development, validation, and delivery of new information and technologies for managing pests important in desert, vegetable, and melon crops that reduces grower reliance on high-risk, broadly toxic pesticides without sacrificing yield quality and profitability. All right, take it away, John.
Okay, first of all, is Gordon still awake? Oh, okay, we're good to go then. Yeah, good morning. I would like to discuss with you my understanding of this interaction between Thrips and INSV from a desert perspective. I know very little about INSV. All I know is what I've seen since March. I know a lot more about Thrips. I've been working with Thrips for the last 25 years here in the desert, and in particular, trying to understand their biology and ecology as well as their management. So I'm going to share with you some ideas I have on how you might rethink Thrips management now that things have changed in the last few months. Thrips management, from my perspective, and I think from yours as well, prior to this recent invasion was pretty simple. You were focused on preventing cosmetic scarring on lettuce. That was the limiting factor to thrips. That was the economic importance of the thrip, was causing economic scarring and some other things. And that could be done, that could be taken care of, as obviously it's been being done for years through PCA management programs. You know, this is one of the typical slides I used to show, the keys to thrips management in the desert. Early detection of thrips population, you really want to scout them thoroughly, see when they're first showing up. Treat as needed to prevent scarring. We try to develop thresholds, but it's a moving scale. But so basically the whole purpose of treating and management was to prevent that cosmetic injury. Use insecticides at effective rates, by ground when possible. Pretty straightforward, pretty effective for the most part by the desert PCAs. Of course, that all changed last March. When we'd been hearing about Salinas the previous season and the issues they were dealing with, and then it showed up here in the desert in early March. There were spot reports throughout the area. We saw some key areas. I think overall, when you take in the entire desert region, it was probably very low incidence overall. And to top that off, it was a relatively light thrips year, as thrips go in the desert. So did we dodge a bullet? We'll never know. But that was something that was real important. When this happened, I started to get very curious about this virus and about this interaction with thrips. So I started to do some survey work out east, eastern part of Yuma. And this is just a graph that kind of shows you the magnitude and the distribution spatially from basically almost to Texas Hill to about Welton, where we were seeing viruses, at what incidence, and these were just simple measurements I was making in the fields. And you'll note that big peak, that was in the Tacna area, and that's, Stephanie described some of the areas she worked in, collected from, that's where we've been collecting from. That seemed to be the hot spot overall. But it was a short window of time. I mean, by April 13th, everything was gone. At least these fields that we were monitoring, the season was over. So we didn't get a really big chance to see what was going on, but we knew it was there. So the questions come up several times since then, and it still comes up. What are the sources of INSV in this particular situation? A lot of people, including myself, speculate that it probably came in on infected plant material or infected thrips coming in on plant material coming from Salinas, given that that's where the major outbreaks were occurring. This just wasn't lettuce. I mean, it could possibly even be brassica transplants. But thrips will, locally, our western flower thrips will readily go to the brassica crops. So it could have, an infected thrip itself could have snuck in on these transplants. So we don't know. We'll probably never know what that primary source that got in. But I'll tell you this. I've been here for 31 years working in this cropping system. In 31 years, I've never seen this virus. We've had stuff brought in. We'd always send it to Steve Koike, and he'd always come back and say, no, we think it'd be tomato spotted wilt virus. No, it's brushy stunt virus or something else. So in my experience, I'd never seen this virus in 31 years. And for it to all of a sudden show up, again, where did it come from? We don't know. Daniel, I borrowed this slide from Daniel, and it's a great illustration of, you got two different things going on when it comes to infection, or as I like to use, spread. You've got this primary infection, I like to call it primary spread, of infected thrips, the adults, remember it's the adults that do the transmission, come from the outside, and they'll hit plants. And from what I saw this year, just last March, it wasn't clumped in the corner of a field necessarily. It was kind of widespread, which again is another big question. What's up with that? I don't know. But this is an important concept. And this is what I call that primary spread. And it'll eventually get to when we talk about management. This is our area-wide trapping network, which many of you know that we've been conducting for the past 8 growing seasons. And we track a number of insects, but one of the things we do track are thrips on these yellow sticky cards that we place strategically in 16 different locations from San Luis all the way to Texas Hill. If you look at this data, you can see that the Tacna roll areas, that's where that big focus was of virus last year, that the peaks are about the same time period. The magnitudes are a little bit different, but you'll see that it was that October-November window. And we didn't see virus until these smaller peaks were occurring in February. And again, if it takes most of the virus I saw was pretty old. It had been there a while. So again, what happened in that interim? Well, we know the cool winter weather, you don't pick up a lot of movement of thrips. And that's kind of what this illustrates. The other thing it illustrates, and again, these thrip counts. So we've got this movement that occurs over the last 8 years pretty consistently. And let me just point out, look at the red line on the bottom. That was last year. Remember I mentioned the thrips, we had a light thrips year? Well, measured by sticky cards, we had one of the lightest years we've ever had in the last 8. Yet we ended up with virus. Again, did we dodge a bullet? Would it have been worse if it would have been like 2017, 2018? We'll never know, but it's very possible. But the question is that there's a pretty big gap between when they peak in flight, at least we pick up on traps, and then when it shows up late, at least last year. But I've got questions about that peak in October, November, is where are those thrips coming from? That's good. we all speculate. I mean, early melon harvest, that's when those first fall melon plantings start to harvest, or they're starting to break them. Some of that late cotton out there starts getting shredded. That could push them. Alfalfa harvest, that's periodical. You know, we saw that all summer. Could be some of the lettuce, brassica crops. Could be desert vegetation. You know, especially out in that narrow Gila Valley there, you've got all that vegetation to the north. In a normal year, last year we've been going through a drought. That's probably may explain why we saw such light captures last year. And then of course weeds. But I think there's something else going to play here too that influences all of that. And part of that could be the cooler temperatures, because that's when October going into November is when we see our first break in the weather. The monsoon's done, all of a sudden we see a break in temperature. It's not brutal anymore, it's pretty comfortable. And as well as the fact that the winds shift. Instead of the monsoon flows, remember we start seeing that change. Everything's coming out of the north and the west. And that's when we start to see aphids blowing in, coming in about the same time period. So a lot of questions here. Don't have a lot of answers for you, but a lot of questions onto what's going on there. So what I've been, and again, to try to answer some of these questions, I've been involved this last summer in trying to determine what's going on in the non-lettuce growing period, basically from about April till 2 weeks from now, when we start putting produce back in the ground. And so that's kind of what we've been looking at, is what are thrips abundance on some of these crops during the summer? I think we all see them, but what does that mean? So this is just an example of one of the locations. This is the Tacna location where we had 2 infected fields, and we selected fields all around them within about a half-mile area. And those little dots, little circles, that represent where I sampled for thrip. And I used the same sampling method, this dislodgement method on the sticky cards. And again, 5 sites, biweekly samples. And we were also, I was working with Stephanie pulling crop samples and assisting her with pulling weed samples as well. And you can see the list of all the crops that were in the area. Now, obviously what we saw, the crop landscape in Tacna was quite different than what we saw in the South Yuma Valley. In some similarities and some differences. So let me show you a graphic of just the Tacna site where we saw the initial big spike in virus last year. I don't think it's a surprise that particularly in May, early June, that alfalfa had the highest number of thrips, without a question. I was a little surprised that teff grass, which is kind of a new crop to me, it was grown in that locale. It had quite a bit of thrip on it too. And you can see the mixture of all the other crops. I was a little surprised that the melon crops had lower numbers than I would anticipate, as well as cotton, giving the blooming, the blooms that are available for thrips in those crops. Again, there's just that picture shows you a description of the dislodgement method that we use to capture them. We bring those yellow cards into the lab and identify the species, as well as just get crude counts. But another thing to mention, probably the most important thing that I've learned this summer in conjunction with what Marco and Stephanie just talked about, is the weeds. We picked them up on a number of weeds. Marco mentioned cheeseweed. Now that was more towards April and May, but it was one of the most, we saw more thrips on cheeseweed or malva than we did on a lot of the others. Same with goosefoot. Goosefoot was loaded with thrip. But what's really important to note is that in every case except for one, that being the salt bush, which was pretty light, we found larvae. We collected larvae on all of these weeds. And that just tells you that it's a reproductive host. Those weeds serve as a reproductive host for thrips. Again, you have to have larvae development to acquire the virus to ultimately move and spread it. So that's pretty important for us to know, that these weeds can be a factor in this green bridge we're talking about from sustaining that virus from late spring to the following fall. Same can be said for all the crops we sampled. They all had reproductive populations. So that's why we have such big thrip numbers in the desert, because they're year round. Stephanie mentioned the surveys for the alfalfa and cotton and melons. I'm not going to say much. It's like trying to find a needle in a haystack. The density of leaf material out there relative to what you can possibly get a very, very good sample. She's absolutely correct. transmission studies are going to have to be done to determine whether these are significant hosts. I personally don't think so. I think if they were, we would have seen tomato spotted wilt virus, assuming they were hosts years ago. We've never seen either of them. So my guess, and it's pure speculation, is that these three crops probably aren't drivers. I'm probably wrong. We'll probably get clobbered coming into the next season, but I'm hoping that's not the case. So let's talk about cultural management. And that all sets up this whole idea about how can you modify or improve your farming practices to manage this primary spread? Because now we're talking about the primary spread. Once they're infected and they hit the plant, Daniel noted that you got about 5 minutes. They can transmit it in as short as 5 minutes. Pretty hard to stop that when you've got them coming in. But there are things you can do to avoid or reduce the availability of those thrips. One of them is, timely and rapid harvest. Again, that's a marketing decision more than not. It's a tough one to deal with. I hear complaints from PCAs all the time about the field was cleared 2 weeks ago and it's still sitting out there. The problem with that is just thrips continue to build in that process. That goes with prompt destruction. Once that field is finished, it's best to get in there and just destroy the crop residue as quickly and as efficiently as you can. The same reason, you leave it out there for 2 weeks post-harvest, that you're generating more thrips, the potential to move. And then finally, I think this is probably the easiest thing to do, and it's what's being done every summer in Yuma, and that is clean culture. Both weed control on ditch banks, in low-lying areas, roadways, edges of fields. This just driving around, and trust me, I've been looking for it this summer, but just driving around this summer, it's extremely clean. I'm real impressed at how weed-free our growing area is, at least speaking for Yuma in particular. And that's important because, again, you're lowering the potential for the vector and potentially the virus too, but certainly the less vector you put into the system, thrips, coming into the fall, potentially you can probably avoid or reduce primary spread. Summer fallow management, which is going on right now, it's particularly important with purslane. Tends to come up after the first pre-irrigations. Growers are doing a great job about getting out of that. Volunteer melons, we had a lot of melons this year, and you can't find a lot of them out there, but occasionally you do. And the problem with that is you've got 20-acre block of volunteer cantaloupes with thousands and thousands of blooms. Even though it's not a host for the virus, as far as we know, it's a source for thrips. And so if you build up thousands and thousands of blooms with even one thrips per bloom, you can see that the potential distribution or dispersion once that field is dished or burned down onto the next adjacent crop. Rainfall, we know rainfall is suppressive. It's a natural suppressive agent for thrips. We've noticed it over all the years. And we know that sprinkler irrigation probably plays a role in suppressing thrips as well. This is going to be a question on the quiz potentially. I'm not going to show you any data, but we've actually done studies where we've looked at duration and frequency of sprinkler irrigation. And what we have found is that you can see 40–50% suppression of the thrips for a couple of days after a good sprinkler irrigation. Not enough to control or prevent cosmetic injury, but you are getting some additive impact from the sprinklers. So just keep that in mind as you go through the season, that sprinklers might be your friend, and particularly if you can chemigate in the process, which is equally effective as foliar sprays based on a lot of the work that we've done. So that's that primary spread. Again, you got thrips coming from somewhere. You don't know if they're infected. There's not a lot you can do. So the point there is to reduce the numbers that could potentially come into the system. I think where we have a chance locally is with secondary spread. And that's, as Daniel described, that's where you've got the reproduction, the larvae acquiring the virus and then moving off. They're fully infected and start to move it on, either move to another field or move within the field to increase that. And that's where, to me, and again, this is just not knowing this virus. I've never even experienced it other than what I saw last March. To me, this is where I think we have a chance, if that virus becomes established here, is in minimizing that secondary spread. Again, if you've got about, depending on temperature, 4, 5, 6 days of larval development per second instar, if you can get in there and whack that, you've broken the virus cycle, because it is transstadial development in the adult. So that's That's in theory, and I don't need to tell you guys what the products are to control thrips. Radiant and Lanate for the last 17 years have been the products of choice. And the amazing thing is they still work as effectively now as they did then. This is data we collected over the last 17 years in efficacy trials. We've seen no degradation in efficacy based on common methodologies year after year after year. We're still seeing the relatively same response to thrips control with these two products. Now, this is larval control. But the bottom line is we've sustained efficacy. And part of it is, I think, our summer cropping system. After the produce season, where are these thrips going? Into the cropping landscape and weeds, where they're not being sprayed with either of these two products. So, but that just goes to show you, part of it is the fact that these are obviously the two products that are used by the industry based on our surveys, our lettuce surveys. And I think this also goes towards resistance management. The fact that, for every 2 radiants, you might be using 1 Lanate. And part of that's label restrictions. But part of it is responsible use by the PCAs. So you can see the major 4 products that are used for thrips management have been used for thrips management. Is that going to change? Should it change for us here in the desert? Well, let's talk about that. This is basically, and I've showed this, this is the most updated recommendation list. It kind of, based on our research experiences and discussions with you guys, where products fit. And you'll note that first 4, those are the products that probably, and again, they kind of mirror the use statistics, more so Lanate and Radiant, but that's the products that we generally think have the best choice to pick up an adult or pick up a larvae in the field. The question I've been debating with the last few years, and I continue to collect data on these other products, is what do these other products play in the role of thrips, and in particular now that INSV could potentially be a problem or a threat for us in the desert. So I can show you tons of data. I'm not going to drag you through all these efficacy trials, but for each one of these products, I can show you lots of data. What I decided to do was to go back in time into my database, and I pulled out studies and generated percent control. 132 trials since 2003. I went in and looked at the data, and for each one of those products where I could, in that little column with the numbers, that's how many trials I actually used in developing these averages, this average percent control. I went in and looked at them individually, and you can see for the most part, and this is the adult. We don't have any really strong adult materials, and that's why this primary spread could be challenging. But something like acephate and Lanate, the old hard school contact OP carbamates, those seem to be the most effective against adults. Obviously, Radiant, part of the Radiant thing, maybe we might be cycling them out in the process. And then Torac's one that's not used much, but at least it does have good contact activity comparable to some of the other products on this list. But you can see where they all fill. I put the solid red line as 50% as kind of the median. So you can kind of look and base that on yours. So you can see all the products and where they fall. It's funny, Movento down at the bottom has absolutely no adult activity, but you wouldn't expect to. It's essentially an IGR, so not surprising. When you look at the larvae, it gets a little more encouraging, I guess. You've got your Radiant, your Lanate, your acephate, your Thorax actually got pretty decent activity. One that surprised me, the more I've looked at it, is Belief. It's actually got some decent suppression at this point, suppression activity against the larvae. As does Sequoia at 5.75 ounces, although we've only had one look at it, and that was it's on the label. It would be an expensive application, but again, you can see that relative to some of the others, it actually has some suppressive activity. And then the others fall well within that 40 to 50 range. So again, the question is, and it's the question that I've been juggling in my head, is what role do some of these lesser products or more suppression-based products play in an overall management program? And I'm going to discuss that for you here in a second. So here's how it falls out for me. If you were to ask me where do I use my products, this is probably how it would fall for me. Those first 3 are probably your hammers, And then in order of importance would be the next group, your rotational alternatives, without a doubt. And there's other roles that we'll discuss here in a second. One other question I've been asked in the last couple months is, mainly it's coming out of Salinas, is will these soil systemic neonicotinoids control thrips? And we've asked that question before, particularly back in the day. Now we're going back to 2003, 2004. We were looking at Admire Pro, and we looked at 6 wet dates. We sampled them thoroughly. And what we found was the only time Admire seemed to be effective was right at the first essential sampling. That at 3 to 4 leaf stages lettuce, you'd see some activity and then it'd be gone. And it was most effective on the September and October wet dates. Part of the problem with that old, and again, it's an old data set, is that we were looking at 16 ounces of the 2F formulation, which is a quarter-pound AI. Now everybody's using the Admire Pro and the generics at 0.375 pounds AI. They've increased the rate 30% because they can, because it's cost effective to use that higher rate. The question we have going into the fall is we're going to go that higher rate and see if we get a bump, see if it improves it. Of course, NIPSIT's been asked about. We've seen some suppression with NIPSIT. Again, 14, 20, 24 days, you'll see some suppression of the thrips. But again, beyond that, it's gone. Problem with these studies, we did those in March. We were so far outside of the planting window that we really need to do it this fall to see in a normal planting window, a real planting window, if it's going to have any effectiveness. That's the question we have now with the neonics. We're going to try to get a more clear picture as we go forward this fall. So let's talk about everything that I've experienced in the last 6 months in terms of the thrip, from the thrips perspective, and what I've learned from Daniel and Steve Koike and the others and people from Salinas. What I understand about both of them, how does that affect us, and how might you rethink how you approach thrips management this upcoming season? Well, first of all, I think it's a no-brainer. The sanitation, that cultural management is essential. We preach it all the time in IPM. Sanitation, cultural management. And I think people just kind of take it for granted. I know I even take it for granted. I just say it because you're supposed to say it. But I think in this case, it's real meaningful. The cleaner you keep the culture from alternate hosts, the less numbers that you could potentially infest your crops. I think that's going to be important. I think early detection is still going to be critical. Getting in there, and instead of just looking for armyworms and loopers, or just looking for whiteflies or aphids, focus a little bit more on thrips. And like I've always said, a few probably means a lot. And I'll put that into perspective for you. I'll show you some old data, but I don't think it's changed. We did some work years ago where we were actually trying to determine, we were comparing sampling methods, but we were also trying to determine what was actually on a plant, a lettuce plant. And we were washing them. We would cut the plants, put them in soapy water, wash all the thrips off of them, and then strain them out and collect those larvae. Well, you can see that in that February window in 2 independent wet dates, plantings, we exceeded 1,000 thrips per plant. And that's a combination of adults and larvae. You don't see 1,000 when you're looking at those plants. You do not see 1,000 thrips per plant, but trust me, they're there. So basically what we have found, we took all of the data and we correlated it, is that you're only seeing about 12% of what's there. Maybe when the plant's smaller, you might see 30, 40, maybe even 50% of what's there, but you're not catching everything that's there. It's because of their feeding behavior and their cryptic behavior on that plant. So you really have to be conscious of what's there. And so again, I've always tell people, if you see 1 or 2, chances are there's 10 or 15 on that plant. And that's important when it comes to virus transmission, because it only takes a couple of thrips to transmit that. If we want to suppress that nymph population, if you start seeing immatures, you really want to get on top of it. I think earlier spray timing, I think one of the take-home messages that Daniel left was that, the earlier the better, because the earlier the plant's infected, the longer it has to express those symptoms and become unmarketable. So the longer you can delay that infection, and it's no different than what we see with cucurbit yellow stunting disorder on our viruses. The longer we can delay the whiteflies from infecting it, the better chance we have of harvesting that crop. No different. So anyway, these I think that, you know, given the fact that we don't know where this stuff's coming from and what we're seeing so far this summer, there's not a lot of virus, at least we're not finding it out there, is that you might even consider a preventative treatment on transplants that are originating from Salinas, knowing that that's where the issue is. Salinas. If you've got transplant, you might think about coming in at stand establishment and hitting it with a thorax or hitting it with a lani, just to kind of knock it off the top. Because of that, now you've got primary spread coming into the area. That's a consideration you can think about. I think there are high risk areas to consider very similarly, and that is that and you might want to treat some seedling crops, direct seeded crops. If you're near fields where there are transplanted crops around you, whether they're brassica or lettuce, knowing that those thrips could potentially move onto your crop, not knowing whether they're infected or not. Certainly in areas where we saw INSV last year. Stephanie showed you, we've picked up virus all summer and it could still be hanging on, even though we can't detect it at our low sample size. There could certainly be virus floating around out there. I think, you know, there's a lot of talk locally. I'm hearing some of the growers talk about incorporating Torac and Lanate into their chemical thinners and their smart sprayers. That's that very first shot. It's a free ride. And you can actually, again, nip that population in the bud. That's the way you guys operate anyway, is with most pests, is when they show up, you want to nip that population from really going exponential. Because as you well know, thrips will go exponential if they're left untreated. Focus on that secondary spread. And I think chemically, that's where, you know, again, chemically, it's hard to stop the primary spread a lot of times because of the weakness of most of our compounds against adults. It's not like you're going to go out there and kill 99% of the armyworm with these thrips materials. You do a good job. But you really want to prevent those larvae from developing. And again, you've got a window of time within that life cycle to do that. I think that's critical. And of course, you guys have your own tricks on applying products and how to use them. And again, you just want to use those effectively. And then again, consider using Torac early. Save your Lanate, save your Radiant and your other products. Tighten those spray intervals, particularly in the spring when those potential populations seem to want to go exponential. Rotate Manectopro and Extrol. I mentioned those two because they're excellent worm and whitefly materials, but they also have thrip suppression. Same thing in the spring. Belief, Acephate, Movento, excellent apicides, but have activity on thrips immatures. So those are some things you might want to consider as we're going into this season. So finally, I just want to conclude with this statement. We have a saying, I teach it in my IPM class here in Yuma, that all IPM is local. And what I mean by that is that the thrips we have here is probably the same thrips they have. Salinas. The virus, according to Steve, is the same. What's different is our cropping system, right? Our cropping system is completely different than the coast. Different growing seasons, different climates, different crop mixes, different landscape cropping and native vegetative landscapes, the way you guys farm, the way you guys manage. So again, what we do in the desert is oftentimes going to be different from what they do in the coast. So I don't want to, I want to make it clear that what's going through my head in terms of long-term management is basically specific for the desert. So that's all I have. Alex, if you want to.
Yeah, thank you very much, John, for an excellent presentation. We have some questions online. The first one from Nick is, what organic material would be the best to use?
Well, there's only really one organic material that will provide the same level of efficacy we see conventionally, and that would be Entrust. I will note this, though, and I've done this, didn't have time to show the data, but I don't know about the cost effectiveness, but every time I've taken a 2% solution of Impede and mixed it with a 5-ounce rate of Entrust or Radiant, it appears to be a synergy of activity. We saw it with Bagrata bug and we see it with thrips. So you can increase the activity of even Entrust if you mix it. And even Impede's got some suppression. The your neem Aza products have some suppressive activity, but it really comes down to Entrust from the desert perspective.
Okay, thank you. Are there any questions there in the room? Nothing. Okay, there's some more up here. So are the thrips dangerous on onion also?
You know, that's a good question. We do find Western flower thrips on onions. I don't know what the proportion of onion thrip to Western flower is, but there is definitely some scarring injury. The only possible virus I'm aware that goes to the onions is Irish spot virus. And we've only seen that once or twice in the last 20 years. And it's, from what I've read, is exclusively transmitted by onion thrips. So I don't know that I have a very good answer for that question.
No, I think that was pretty good. I mean, the onions probably, if you found it, Western flower thrips on onion, it can support that species. So, okay. So another question: which insecticide shouldn't be rotated and why?
Well, I talked a lot about radiant and lanate. There was one I didn't mention at all. That's the answer. But again, Harvanta would be one. Proclaim, that was the question. Those products are excellent lep materials, excellent diamondback moth materials, but they have absolutely no effectiveness against thrips. You'd be sorely disappointed if you applied them for thrips activity. I mean, we've gotten numerous studies that support that. So that's why I pointed out Proclaim. Harvanta falls into that list. Coragen would fall into that list. Those products are excellent for what their targets are, but unfortunately, thrips aren't a target.
Okay, thank you. A question for Robert. Do you have any experience with treatments, especially organics, causing increased movement of thrips within the crop and hence increased infections?
No, I don't have any experience with the virus in any of my trials. It's just about thrips, and I don't know if I can say I've seen any movement. I will tell you this, though, as sort of a related answer, is that I used a bunch of dispersible soluble sulfur this year. It was recommended that I look at it in combination with some of our products that would enhance activity, and the answer is no, it didn't. And I've seen it years ago when we were using other types of sulfur, that you would think that it would aggravate the thrips, make it move, maybe make it flee, but in fact, we didn't see any activity. And dispersible sulfur by itself doesn't have any effect on thrips. Sorry to beat around that question, but that's the best I got.
Okay, no worries. So I think that's all our questions online. I have one question. You've mentioning that the transplants are a pretty important, suspected way that the virus was introduced, and so is that based on kind of just the fact that the virus is a big deal in Salinas and that many of the transplants come from there? Were there any observations in your surveys where you saw it was associated more with transplants?
Yeah, a couple of things. First of all, in 30 years, never saw this virus or even tomato spotted wilt virus. So again, I thought, well, it's not just late, and it's possible it could have been latent out there, but doesn't seem likely. Secondly, yeah, we did see some direct observations with certain areas that we saw infected fields, coincidentally or not. Right across the road was a field of transplants that had originated from the coast, Salinas. So you kind of make that assumption, not knowing exactly if that's the case. And then the fact that these transplants can come infected and or with the thrips themselves. And again, I still don't know why our peak thrip movement based on sticky traps, and we'll get a better feel for that as we go into next season doing plant sampling. But sticky trap catches were in October, yet the virus didn't show up until March. Probably infected in February. That's a big question, a big void in my understanding of the epidemiology of this whole system. So again, I could be absolutely incorrect, absolutely wrong about the association of transplants with the occurrence last year, but maybe not. We'll never know.
Yeah, I think it seems like a reasonable explanation at this point. Okay, so just one last question came in. Diatomaceous earth is being commonly used on organics. Does that have any efficacy for thrip suppression?
Hey, Alex, repeat that question again. I didn't.
Sorry. Diatomaceous earth is being more commonly used in organics, does that have any efficacy for Thrips suppression?
I think if you could get contact activity and get it down to where they are, I'm sure it would be. I've never looked at it directly, but diatomaceous earth is an underappreciated compound. Again, it's all in the application. If you can get it, if it's a dust formulation in particular, if you can get it to where the Thrips are, it can actually be effective. We used to see it in the whitefly days, and we were really pursuing that. And then along came imidacloprid and didn't have to worry about it anymore. So yeah, I mean, I wouldn't doubt that at all. If people are seeing activity, it's probably worth continuing or investigating further.
Okay, so just looking at the answers now, everyone did well on number 1 and 2. Number 3, 72% chose D for which of the following insecticides can be applied to spinach or lettuce to control aphids and also provide suppression of Thrips larvae. Most people selected a true for number 4, 82%, and then number 5, 71% selected proclaim for which of the following insecticides should not be rotated for control and resistance management of Thrips in lettuce. So any comment on that, John?
No, I think that's pretty good, especially being the last speaker. People tend to nod off a little bit. That's pretty good responses. Yeah, and we are a little bit over time, so we apologize for that. But yeah, so that's it for the program. I think as I kind of wrap it up, Sonnet is going to put a notice for a survey that is going on, and so as she does that, I just wanted to thank again everyone for our speakers, everyone for participating, and especially thank Sana and Robert for a lot of legwork with getting this organized. Yeah, so thanks to our speakers.
No, I just want to say thanks to Robert Masson for helping so much with getting the CEUs, and Sonnet, who was just great for the logistics of this event. So, and thanks to everyone for joining us.